including this Registan square, Shahizinda complex, Vivahane, Mosque. This is reflecting the city-rich Islamic architecture and cultural legacy. Many tourists also visiting in Uzbekistan because there's, if you're visiting in Uzbekistan, you find a lot of historical buildings such as Samarkand, Bukhara, Tirmiz, and Khorasan. Bukhara is one of the most ancient cities in Uzbekistan, known for its preserved mosque, madrasas, and minarets. It was important stop of the Silk Road and remains cultural hub today. The architectural masterpieces such as Kalyan Minaret and Mira Amar Madrasa are symbols of Islamic heritage. And my hometown is Sukhandaria. Sukhandaria also so beautiful. This is province. Each province is a different culture, and also this is also traditional dress also changing, food also changing. This is also in our country in the Sukhandaria province, and the woman is in the clothes. Then in the Uzbekistan also, famous art in Uzbekistan centers on traditional handicraft, particularly Sudana embroidered ceramics, such as in the Rishtan, gold embroidery, and wood carving, jewelry making, and other. And Uzbekistan also very famous in the Uzbek cuisines and also delicious food, such as in the palau, our national in the cuisine. Also manti, samsa, laman, and also bread, also different type, small bread, bigger bread like that. And nowadays, Uzbekistan also so beautiful and modern Uzbekistan. Many tourists also visiting in the sea, in the magic city. This magic city also so beautiful. I hope if you're organizing next year international conference, you welcome in the Uzbekistan. Then nowadays is salinity is major problem in the world. How can affect in salinity? Salinity also decreases plant growth yield, decreases also nutritional status of plant. Also the salinity induced in the osmotic stress and also decreases in the rate of seed germination and also yield. Then what is biochar? Biochar is a carbon-rich material that is made from biomass raw. A thermochemical conversion process known pyrolyzed biochar. A natural material obtained by pyrolyzing organic biomass such as plant straw under limited oxygen. This is the F300 and 1000 grams. Most of these 300, 400 also given in the good yield in the biochar pyrolyzer. Biochar also important role in the improving soil quality, improving in the soil microbial biomass. When this applying in the soil, then also improve in the plant growth and physiological properties and yield under drought stress, also salinity stress condition. Then nowadays, a lot of scientists also submitted a paper in the Biochar journal. Biochar journal also high impact factor journal. Within 10 years, more than this 64,000 paper is published about in the biochar. In the Uzbekistan is main problem in the salinity. Most of this in the Uzbekistan land, 44 in the process land also salinated area. Then in the first time we also this experiment doing this in the Aral Sea in the dry bottom area. Then we applied in this in the salinated soil to biochar. What is biochar? Biochar is very rich organic material and organic carbon, and also this rich in the nitrogen, phosphorus, potassium, and also including in the micronutrients such as iron and zinc and molybdenum. This is also beneficial effects zinc, molybdenum to improve in the plant growth. In the when we applied in the biochar treatment, also beneficial effect in the plant growth parameters of alfalfa, amaranth, maize, and under such stress condition. In the compared to control, also the similar results show that when we use it in the biochar treatment, significantly increases in the plant height, shoot fresh weight, root fresh weight, biomass, and maize plant under saline stress condition. When we applied in the biochar treatment, also this significantly enhanced uh, root morphological traits of alfalfa amaranth maize plant, such as in the significantly increased in the total root length, root project area, root surface area, root volume, root diameter, and the saline condition. This is current situation of Eastern Aral Sea. Soil degradation in the Republic of Uzbekistan mainly caused by salinity, 54%. When we starting in the our experiment, this is this area, because this area is also drought stress area and also salinity stress area. Then biochar also this positive correlation in the photosynthetic pigments such as chlorophyll A, chlorophyll B, total chlorophyll, and carotenoid content. Biochar treatment also significantly enhanced in the total chlorophyll content and the chlorophyll A, carotenoid, and also chlorophyll B content in the alfalfa, amaranth, and maize under saline stress condition. Then also biochar not only this beneficial effect in the these photosynthetic pigments, also these improve in the water holding capacity and also in the beneficial effect related to water content. Biochar treatment also significantly enhanced in the related water content in alfalfa, amaranth, maize plant under saline stress condition compared to control treatment. After harvested this experiment, we also this analyzed in the soil nitrogen, phosphorus, potassium, humus content. When we use it in the biochar treatment, also this significantly enhanced in the soil nutrients such as nitrogen, phosphorus, potassium, humus content. How can effect in the biochar in the soil enzyme activities and the soil stress condition? After harvested, this is also in the alfalfa amaranth maize plant harvested. We are also analyzing in the enzyme activities such as catalase, invertase, urease. Then compared to control under stress condition, biochar also significantly enhanced in the catalase, invertase, urease enzyme activities in the soil treatment. Then biochar treatment also in the positive correlation with in the plant growth parameters, also in the photosynthetic pigments. This is FEDMAP also showed that in the blue color also showed in the positive correlation and the variable treatment. Then in the we also this working in the saline stress, also drought stress, because in the saline stress also drought stress also major problem in the Uzbekistan. What is drought is effect in the plant. Drought stress also in the decrease in the germination rate, also this decrease in the photosynthetic pigments, and also year by year also in the agricultural crops lost the yield year by year. We also doing in the our experiment used in the biochar and also Arbusca mycorrhizal fungi. Arbusca mycorrhizal fungi also is improved in the plant nutrition delivery, plant growth, productivity, soil quality, also fertility, abiotic stress, and the tolerance also resistant to pathogen. Mostly this when this applied in the this in the Arbusca mycorrhizal fungi, also this in the improve in the nutrients such as nitrogen, potassium, phosphorus, calcium, and magnesium. And we also dealing soybean food further in future. Why? Because in the soybean also high rich in the protein content, and also oil and also carbohydrate, micronutrients such as phosphorus, potassium, nitrogen, and micronutrients such as iron, zinc, magnesium. And soybean also used in the food industry, in the textile industry, in the chemical, and the engineering industry, in medicine, and others. And biological fixation supply nearly 200 million ton nitrogen a year in the world. Liquid rhizobium symbiotic fixation occupy 65 and 30%. Uh, with this soybean, not only this high rich in the protein, also this soybean improve in the soil in the properties because in the soybean also this symbiotic performance within the Bradrhizobium japonicum also this improve in the soil in the properties. Then will be this user test in the our experiment biochar and also beneficial effect plant height, shoot drive it, root drive it, nodal number in the soybean plant under drought stress condition. Also similar results show that will be this alone used in the arbusca macarizal fungi also improved in the plant growth parameters in the soybean plant under drought stress condition. Compared to all treatment, combination of biochar and arbusca macarizal fungi also this beneficial effect in the plant height, shoot drive weight, root drive weight, nodal number in soybean plant under drought stress condition. And when we use it in the combination of biochar and arbusca macarizal fungi, compared to all treatment, also significantly enhanced in the root morphological traits such as total root length, projected area, root surface area, root volume, root diameter in the soybean plant under drought stress condition. Then biochar and arbusca macarizal fungi alone also this improved in the chlorophyll content, in the nitrogen, in the carbon content in the in the soybean plant under drought stress condition. But will be used in the combination of biochar and arbusca macarizal fungi significantly enhanced in the this soil nutrient, plant nutrients, also in the chlorophyll content under drought stress condition. After harvested, our experiment we also this analyzed in the these soil enzyme activities such as alkaline phosphomonoesterase, dehydrogenase enzyme activity, also fluorescent diastase enzyme activity. Compared to all treatment, dual application of biochar and arbusca macarizal fungi also in the improved in the alkaline phosphomonoesterase, dehydrogenase, and fluorescent diastase enzyme activities under drought stress condition. When this applied this in the arbusca macarizal fungi and also biochar treatment also improved in the this arbusca macarizal fungi spores number and microbial biomass because in the biochar also this beneficial effect in the soil in the microbial biomass. We also, when I was conducted my research work in Indian Agriculture Research Institute, IRA, because this time is my one project, Uzbekistan project, and continue in the turmeric plant. I also working in the in India turmeric because in the mostly this in India use it in the turmeric plant for in the spice food or some in the medicine. And in our experiment, this is in the biochar and arbusca macarizal fungi alone, also and improved in the turmeric plant height and also leaf number, shoot biomass, and also this shoot and root length compared to control treatment. This is with this used in the combination of biochar and arbusca macarizal fungi, also beneficial effect in the this rhizome and also rhizome biomass significantly enhanced in the combination of biochar and arbus kamakarisal fungi. And biochar treatment and alone emit also beneficial effect in the photosynthetic pigments compared to in the control treatment. But compared to all treatment, combination of biochar and arbus kamakarisal fungi also beneficial effect in the photosynthetic pigments such as chlorophyll A, chlorophyll B, total chlorophyll, and carotenoid content. And after we also this the effect of biochar and arbus kamakarisal fungi and combination of biochar and arbus kamakarisal fungi also was studied in the turmeric plant in the physiological properties such as photosynthetic rate, stomatal conducted transpiration rate, and what you use in the FSL. Compared to all treatment, combination of biochar and arbus kamakarisal fungi also this beneficial effect in the physiological properties in the turmeric plant under drought stress condition. After we after we harvested in the this turmeric plant, we also this analyzed it in the soil nutrients and also soil enzyme activities. This is compared to all treatment. When we use it in the combination of biochar and arbus kamakarisal fungi, also this beneficial effect in the nitration and phosphorus and potassium content in the soil. Also, this similar result showed that when we use it in the biochar and arbus kamakarisal fungi together, significantly enhanced in the alkaline phosphomonoesterase, dehydrogenase enzyme activity, and fluorescent diester enzyme activities. Conclude. Okay. Then I also feeling science without borders because I also visited many countries in the this in the Germany and this Chile, Norway, and others. Then in the June I also visited in the Chile. Then should be the science also visited any country, also learning, also this research work. Then I also acknowledgement in the my special thanks to Professor Amar Garg, Dr. Joginder, also inviting me this wonderful this conference. I also special my thanks this organizing team. Thank you very much, all of you.
Yeah, thank you very much for your excellent presentation, and of course you covered a lot of information, various research innovations and invention, particularly the biochar shows how it's very beneficial in maintaining the soil health, also the nutrient enrichment, and also to tolerate the unfavorable condition, droughts and stress. So that is a very good piece of work. Thank you.
Thank you.
So yeah, on behalf of the organizing committee, I take this opportunity. Thank you once again. So yeah, just I forgot to introduce my co-chair in the beginning of the session. He is Dr. Aucha, is a professor of botany, Vidya Physical College, Varanasi, district Pune. This college is affiliated to SPP University, Pune. He is co-chairing with me. Of course, you know me, I am Professor H. Shivanand Murthy. I am presently working as the Dean and Chair Professor at Neotia University in West Bengal. I also served as earlier as the Registrar, Dean, Director, Karnataka Veterinary and Animal and Fishery Sciences University. So more than 45 years of experience in research and development, higher education, and also in fisheries, animal husbandry, and agriculture. Thank you. So we move on to the next lecture by Professor Dr. V.K. Sharma, is a senior scientist, division of bioenergy. Yeah, bioenergy then bio refinery and green chemistry, Inia Research Center, Triscia, the Rotandella, Italy. And in fact, briefly to introduce Professor V.K. Sharma is a renowned scientist and also worked in various capacities, and he worked in Italian Commission and of course new technology, sustainable economical development. He has been recipient of the about 3 gold medals, then lifetime achievement awards, the member of various organizations, and is the editor and co-editor of several international journals. He has published more than 185 papers, 5 books, and is a works also national consultant, member of UNESCO, ENDP, ENDO, and ICTP. So with this brief background, I invite Dr. V.K. Sharma for his keynote address.
Thank you, Mr. Chairman. ABCD. Second last. First of all, I would like to put on record my sincere thanks to Professor A.P. Garg, and then, of course, Dr. Julinder Singh, who has invited me. It was just by chance that I was here in India, so he asked me if I can come and participate at the conference. So it was a great pleasure for me to meet all my colleagues and friends. So I have a lot of information, but because of the time limitation, maybe I will run little bit fast. So my topic is far away from the theme of the conference. So anyway, it covers that energy is the main source for every type of activity. So my paper will be focused on clean energy technology, and that will for the sustainable development in the future. Yeah, this is just as we face growing environmental challenges, innovative solution and advances in agriculture and biodiversity conservation are crucial in sustainable development because they support not only food security and economic growth but help preserving the environment as well. Advances in agriculture like precision farming, innovative concept of agro-photovoltaic can help increase yield while reducing chemical input and environmental damage. Biodiversity is essential for providing ecosystem like pollination and pest control, which makes agriculture more resilient, especially in the case of climate change. So, for the definition of the sustainable development, so the most acceptable definition is sustainable development defined as an approach of developing a growing by using resources in a way that allows for them to renew or continue to exist for others. Is probably the most important thing that a common man can hope for. For rights and well-to-do people, it is a bit possible to sideline this issue, but the common man will be affected the most if we fail to implement this. So, variety of scenario can be used where sustainable development brings about good to the common man. Use recycled material or renewable resources is an example of sustainable development. So, where environmental pollution is concerned, energy efficiency and utilization of renewable energy sources seems to be a better route to follow. So, the International Conference on Advances in Agriculture, Biological and Biodiversity Conservation for Sustainable Development will provide a forum to the best talent and academia and research institutions, industry expert and executive to collaborate and address the current challenges to seek new windows for the discovery and exploration and to support the evolution of science, engineering and technology. For sustainable leaders. The conference is aimed to provide support to academia and industry. And after this, there is a group of tertiary technological and research science. We hope that the sharing can there is some multiple types. Will be a rewarding experience for all and provide better opportunity for vibrant global production. Thank you, sir. So I do hope that eminent speakers with their high caliber presentation, in-depth and very pertinent observation will guide the faculty and participant, ensuring the importance to promote the multidisciplinary approach which is so important for the sustainable development of a nation. I extend my warm greeting, heartiest felicitation to the organizer and the participant, and my best wishes for a successful event. So a few words about my institution. So NIA is the Italian National Agency for New Technology, Energy and Sustainable Economic Development, with its head office in Rome. Focus are on areas such as energy, environment, production, technology transfer, and sustainable economic development. The institution is not only that with highly highly skilled laboratory, experimental platform, and cutting-edge facilities. Career transition aiming to improve the integration and for the industry and various across the energy value. In this context, Kenya design and develop proper solutions and applying technology for great smart city and smart mobility. Kenya currently operates a center for nearly 2,000 center and immense research laboratory. Because there are kinds of budget from more than nearly 5 million years. At the international level, Kenya participates in technological platforms and networks like Terra, European Energy Research Alliance, TECRA, European Climate Research Alliance, Regular Mediterranean Association of National Institute of Technology, Enterprise Alert Network, network of success supporting business and impression for SAPs and the numerous technology and institution from India as well. So this is a slide that will say a few words about the energy shortage. So green energy is treated with the last amount of adverse environmental effect possible, such as global warming and climate change. Renewable energy sources, including hydro, geothermal, solar, and wind energy, are developed and marketed as substitute with that contribute little or nothing to the global warming process. Owing to the fact that some nuclear technology produce far less waste than coal and oil, even nuclear energy is sometimes regarded as green energy source. Nuclear and renewable energy are two viable alternatives to fossil fuel-based energy generation. So new energy, which one? So it is unanimously recognized both by the scientific community and the politicians worldwide that natural resources such as nuclear and renewable energy are the only two potential options to meet long-term energy survival of mankind. Nuclear energy, that is via both fission and fission nuclear reaction. Some also consider nuclear power and renewable power source also that is controversial. So far the renewable sources are concerned, that is solar, wind, hydro, geothermal, biomass are developed as low or non-carbon alternative to conventional energy sources. So although fossil fuel represents the main source of global energy, covering almost 80% of the global energy consumption, needless to say that the option to consider increased use of renewable sources should be looked at as an important pillar to decarbonize the energy system and instrument for the socio-economic development, eradication of poverty, unemployment, and rural development. So this is a traditional classification of the renewable. From the slide, it is very clear that the main source of all type of energy that is available on the earth, that is sun. So the energy from the sun can be used in two different ways, that is direct use and then indirect use. So where the direct use is concerned, that is through solar thermal or the solar photovoltaic, and the solar thermal can be further considered at three temperature ranges. That is low temperature, medium temperature, and the high temperature. When we say the high temperature, that means when the temperature requirement is more than 300 degrees Celsius. Whereas if we consider the indirect route, that can be done through the wind energy, hydro energy, biomass, and others. And so far the use of biomass is concerned, that is through two process, that is thermochemical process and biochemical process. So now I will say a few words about the solar thermal. Solar thermal collector at low, medium or high temperature have been used for many years for both commercial and industrial application, including domestic or industrial hot water system, solar cooling and electricity generation. It is important to involve the policymaker, representative of the solar thermal sector and other industrial sector, highlighting them the contribution the solar heating and cooling sector can bring to a decarbonized and decentralized energy sector. So here I must say that so far solar thermal is concerned at low temperature, there is nothing much to do so far R&D is concerned, because the need of the hour is to implement the result that have been obtained. So solar heat when applied in both the pulp and paper sector has significant potential to accelerate the decarbonization of the industrial process. Also there is enormous potential for cooling systems that use solar energy, both solar thermal and PV driven solar cooling and air conditioning system. Solar thermal driven system customize less conventional energy and use natural refrigerant such as water and ammonia. Can I have the water, please? Yeah, I just need some water. Solar thermal cooling is still a niche market. However, awareness of small to medium solar thermal driven system is rising and several step projects are in progress. So this is development of the renewable energy not only affect environmental aspect but also create jobs. According to a survey by Arena, renewable in 2023 had employed over 16 million people. As regards specifically the PV sector in 2030, there were approximately 7.1 million people employed in this particular sector, of which approximately 4.6 million in China, followed by India with approximately 3 lakhs 10,000 new employees. So, research and development topics of interest in solar thermal. In brief, research and development topics of interest in solar thermal and low temperature application are research and development of energy storage for solar energy application at medium temperature, research and development of hybrid system using solar, wind and biomass, develop a toolbox including the basic component for the construction of small to medium size concentrated system multi-generator power plant to be integrated in building. The goal should be to combine and optimize innovative subsystem for the efficient capture of solar energy to generate electricity, produce heat and cold, drive thermal process such as desalination or sterilization alone or in combination with a backup. To deploy technology through technology optimization, integration and validation, use case study analysis and impact assessment through small scale cost-effective unit of polygenerator concentrated solar technology. So these are the R&D topics of interest that must be considered. So now the part 2 is the generation of electric power using solar energy. So you can see that tracking concentrated system or non-tracking concentrated system can be used to generate electricity. So there the concentrated solar power plant produces electric power by converting the sun's energy into high temperature heat using various mirror configuration. The heat is then channeled through a conventional generator. The plant consists of two parts, one that collect solar energy and convert it to heat and another heat that convert heat energy to electricity. There are, as shown in the figure, there are four main tracking solar concentrated technology familiar, namely parabolic truck pump collector, parabolic dish, linear Fresnel reflector, and solar tower. Classified by the way they focus the sun rays and selection of the technology used to receive the sun energy. So the ANIA concentrated solar energy program, by using parabolic trap collector, ANIA has developed an innovative technology chain based on a molten salt mixture functioning as heat transfer fluid and thermal storage. Compared with diathermic oil, use of molten salt mixture allows the power plant to operate at increased temperature, thus resulting in significant benefit to the plant operation, safety and environmental friendly impact. Other considerable advantage is the possible integration of the solar plant with a conventional thermoelectric plant and an efficient thermal energy storage and increasing the dispatchability of the electric power produced against the unpredictability of the solar source. So these are the innovations that have been done at NIA. So NIA introduced a new fluid heat carrier, that is molten salt, that is a mixture of 60% sodium nitrate and 40% potassium nitrate in order to increase the operating temperature and the possibility of storing heat. Another innovation of NIA is the design of a new type of concentrator based on thinner layer that saves construction and installation cost. The use of large heat storage. Another innovation in the Archimedes project enables the plant to provide heat to the steam generator at constant rate of 24 hours a day, regardless of variation in the solar energy availability. 5 megawatt capacity solar plant costing nearly 60 million of euro has the unique characteristics of to collect and conserve thermal energy of the sun for many hours, thus enabling the plant to generate electricity both during off sunshine hour and the overcast time. This proves that the cost for per kilowatt hour produced is nearly 5 or 6 times more compared to the cost of energy generated using conventional fuel, but with 2,100 tons of oil saved and 3,250 tons of carbon dioxide emission reduced in a year, the present achievement is certainly an important milestone.
Yeah, please conclude. Yeah, just conclude. Time is over.
Hmm?
You please conclude. Finish. It's time is there's running short of time. Yeah, please.
I come to the conclusion then. No? So the renewable energy technology are diverse and can save the full range of energy service needs. Various types of renewable energy can supply electricity, thermal energy, and mechanical energy, as well as produce tools that are able to satisfy multi-energy service needs. Renewables is any form of energy from solar, geophysics, or biological sources that is replenished by the natural sources at a rate that equal or concedes its rate of use. Unlike fossil fuel, most form of renewable energy produce little or no carbon dioxide emission. Renewable sources prove to be strategic importance to achieve a sustainable energy supply. Today, low temperature, less than 100 degree Celsius thermal solar technology are reliable and mature for market. So, for generation of electric power using solar energy, novel concept developed and experimented by NIA, that is Archimedes project, combining the best technology of today with that of tomorrow, consisting of a solar field, a storage system, and a steam generator. Solar disk powered by a micro turbine, etc., needs to be implemented on industrial scale. Due attention needs to be focused on the installation of fixed bed gasifier plant of power capacity 20 to 30 and 80 kilowatt electric, most appropriate for the diffusion in the agriculture sector, especially in the developing country. Contribute renewable energy will provide them the portfolio of low carbon technology. Heavily depends on the current economic competition between these technologies. Their relative environmental burden beyond climate change as well as around security and social aspect. Hydrogen is a crucial component in promoting the energy transition. Hydrogen offers a way out to carbon heavy industry, but its contribution to net zero emission will require major investment and coordination. This is just a few words to thank my organization who has given me the permission to attend, come and participate at this conference. So last but not the least, the text used in the presentation from various authors worldwide is duly acknowledged as well. Thank you very much.
Thank you so much, Professor Sharma sir, for giving very good information on energy for sustainable development. You have also mentioned your INEA Research Institute, Italy, and you have focused very good light on global energy sources, nuclear energy, renewable energy sources, and traditional classification of energy. And lastly, you have very good mention energy for commercial application for industrial and agricultural section. On behalf of all organizers and all participants, I extend my grateful thanks. Thank you so much.
So I would like to take another 2 minutes. So I would like to inform the young generation, young researchers who have already done their PhD or they have already completed their engineering degree, that there is a opportunity to visit and avail fellowship from Italy. There is one program that is called that is Training and Research in Italian Laboratories, and that is operated by International Center for Theoretical Physics. So if you go to the site ictp.it, there you can go for search for TAIL program. Okay, so all the information is available over there. So almost all aspects of the science they are covered. fellowship will be provided, and you can avail this opportunity for 6 months to 1 year, and even it can be extended for another 1 year also. This is for the young generation, and then for the faculty who has already completed their 45 years, they can also apply for a program that is called junior associateship, that will give you an opportunity to visit Italy for 3 times in a 6 year period. Each time the duration of the visit will be 90 days, and the everything will be covered. Financial aspect will be covered fully. So I request that this program is also information available on this ictp.it website. Thank you so much.
Okay, thank you. Once again, Dr. Vinod Kumar Sharma. So, just one announcement. There will be 30 presentations, just oral short presentations, and you have to upload all your presentations outside in the laptop. So keep it ready. Okay. So then, yeah, one instruction received from the organizing committee that time is very short. So now I think for all next speakers only 10 minutes. So 10 minutes you have to sum up. So you kindly bear in mind, because otherwise evening we will not be able to finish the other presentations, and there will be a cultural program. So we will not be able to complete. So kindly cooperate. And the next presentation is by Sri Prasad Vista. He is a doctor, Sri Prasad Vista. So he's senior scientist and chief of the government of Nepal, Nepal Agricultural Research Council. Probably you must have heard all Indian Agricultural Council, ICAR in India. Similarly, the Nepal Agriculture Research Council, National Agriculture Research Institute, then National Soil Science Research Center. So that the place is called Kumatar Lalitpur in Nepal. And to say a few accomplishments of Dr. Vista, is the General Secretary of Nepal Society of Science, Nepal, the adjunct professor, Himalayan College of Agriculture, Science and Technology, then focal person, Nepal Seed and Fertilizer Project, USAID project, the national coordinator, International Association of Promoting Geo Ethics, Italy, editorial member of Soil Atlas and Asian FAO, Global Soil Scientist Award, you got then visiting international professor, and most outstanding PA 2023. So with these brief accomplishments, I invite Dr. Vista to deliver his keynote address.
Thank you, Chairman. Good evening, everyone. Respected Chairman, Co-Chair, distinguished scientists here, and all ladies and gentlemen. So I'll be presenting on biochar. So most of the things has been covered by Dr. Dilpuja. So she made me very easier. I'll be presenting some of the things which we did together in Nepal during last 7 years. So I don't need to say more about biochar, because biochar you have already heard from Dr. Dilfuja, and this is a carbon-rich material which is produced from organic biomass through the process of pyrolysis. So pyro means heat and lysis means breakdown. So the process of making biochar is very easy. You know give heat, then the final product, the biochar. And why we need to use the biochar? So it has it's very highly porous. It absorbs water 600 times, 600% moisture it holds as compared to its volume. So and the surface area is very high. So with the very little amount of biochar, you can apply in a very large area, and it holds nutrient, and it retains water for a long time. So in order to slowly release those nutrients, whatever we apply, biochar it plays a very good role, and it is also since it is a carbon rich, so carbon is the source of food for the microorganisms. So microbial diversity as well as population it increases with the application of biochar. So this is one of the thing which just I wanted to highlight. So biochar, generally is not an alternative to fertilizer. It's a soil conditioner. So you can see here, this is the soil only soil, soil with only biochar. If you apply more biochar in the soil, sometimes it gives negative result. So if you apply biochar along with the nutrients, it gives good results. So because as I said, it holds the nutrients. So in case if you apply only biochar, in that case, it holds and the nutrients may not be available to the plants. In that case, these things happen. Okay. So what we did, I am representing on what we did in Nepal. So in 2015 to 2017, we did tremendous work. We did around 244 trials across Nepal, and that was done we tested in 22 crops. So among those 244 trials, 113 trials were in farmers' field, others were in the research stations. In different research stations, we did laboratory experiments. We did glasshouse experiments. Then we did field experiments. So tremendous work has been done in so many crops. These are the crops which we did. So tomato, potato, barley. Just I'm not going to give detail on 244 trials. I'm going to give the summary only. I'm going to present here summary. Some of the findings, what we did, what we got. So this is quantity. How we prepare biochar. This is quantity. This is soil pit king. We say corn ageno. So the biochar prepared in quantity is, if we prepare from here in the soil tin itself, then they have similar, quality. So it's more economical because farmers can make peat by themselves. But the thing is, there is something, science behind the peat. You have to make certain diameter. Then the angle of inclination should be, should be equal with this one because when you light the fire, by the time the air should not enter into that because if it enters, it becomes carbon dioxide or carbon monoxide. Then it emits the greenhouse gases. So this is one of the mitigation options biochar. So that should be taken care of. And one of the things is we have corn cobs. So these corn cobs, after we sell out, then these are one of the best feedstock for us. In Nepal, we don't use anywhere these things. So other things are used for agricultural residues are used as a feed for the livestock and others. And when we did quenching, we quenched with different like with soil also we did, and we used water as well. But we found that quenching with the cattle urine, because in Nepal generally we have a mixed farming system. So every farmer around they have 1 to 2 cattle. So they have cattle urine. That cattle urine, if we use for quenching, in that case, those nutrients in the cattle urine that become the biochar become enriched with those nutrients, and that becomes very effective in case of crop production and productivity. So that was one of the findings. And other things, these are some of the laboratory works, but how the characteristic of the biochar, but whichever we are producing, so it has this much pH carbon content is around more than 70% we got. So cation exchange capacity 72 centimole. And we also analyze fresh cattle urine and we fermented for 96 hours for 4 days. Then we fermented it with biochar. Then in that case, the ammonical content increases around twofold. So we generally recommend them to ferment and use that, biochar. So this has very good effect on soil properties as well in case of soil pH. nitrogen content, carbon content, as well as the potassium content. It increased the soil properties. So we did some work on germination test. We did germination test. Then we did earthworm evidence test. So we this is fresh test. Fresh. So if we swap the carbon, biochar with the cattle urine, in that case, germination is zero. Directly we cannot use for the seeds. So that has to be covered with the soil. Biochar, after apply, you have to keep some around 1 inch soil. Then after that only you have to seed it, so that there is no direct contact with the seed. So otherwise the germination is zero. And here also in other case also, this one is whether earthworm or microorganisms prefer biochar or not. We have tested in different uh, uh, we did a microbial avoidance test, so whether they prefer or not. So preference was very high when we did the experiment with 20% biochar. If more than that, it's not that is good. And this is also another research on Chinese cabbage. This is here also, soil plus cattle urine soft biochar. In that case, the germination is zero. Otherwise, in soil plus biochar, the germination is high. But here, the germination was very less. So this is the earthworm evidence test which we did in different, amount of biochar. So the amount of biochar is 5, 10, 20, 30, 40, up to 50 tons we did. So the best preference is 30 ton per hectare by the organisms. So 30 ton is best, but it's difficult to apply 30 ton at a time. So we say that in the closed cycle, if a farmer could produce around 1 ton or 200 kg in a season, that can be used. And in each season, 3 times, 3 cropping system. So 200 kg, 200 kg, 200 kg, it becomes 600 kg in a year. So over years, 30 ton, in supposing 0.6, 30 tons, it may takes around 30 years, but that lasts for more than 100 years. So this is how we did. So our findings on soil properties, uh, soil properties, with it increases with the pH increases with the increase in the dose of the biochar, and organic carbon also it increases, and available potassium increases. But total nitrogen and available phosphorus, there was no much changes on those things. Okay. Yeah. So my key findings is that we did experiment in 22 crops. So there was no increase in the yield in rice, barley, mulberry, and ginger. And because here rice, the biochar it floats, yes. Then in barley and barley it does not have much, response on biochar or nutrition. So ginger, we got some since Dilfuza also she presented on turmeric, but that was under drought condition. But ours in ginger, when we apply biochar, the moisture remained for long period. So it aggravated the rhizome rot. So that the yield was not much good. And we got 20% increment in these crops and up to 80% on cucurbitaceae family. So this and cabbage, chili, and tea, we got 80% increment in the year. This is what we got. So some of the inferences here. So it performs well in acid soil, and we got better result in the sandy soil, and we recommend it for river basin farms. And it's not much effective in waterlogged condition, as I said. So urine charge biochar. After this is urine charge biochar. So see, after we apply this, and then we have to cover it, and only then we go for planting. Urine charge biochar is one of the good options for nutrient management. For even for the organic farmers, we can go in that way. So we have different opportunities. We have plenty of raw materials that can be prepared. So farmers get so many incentive through by making those biochars through climate change projects and others. So I'm going to the conclusion. So biochar, you know, if a total cost of production for kg of biochar is only rupees 13 in our cases. Rupees 13 per kg. Cost is only that much. So a farmer using 1 ton of biochar, they can reduce the atmospheric carbon dioxide, or they sequester around 2 to 3 tons of carbon each year. So this is one of the findings we did. So in most of the crops, as I said, the yield increases more than 20%. So a household with 2 cattle can substitute 51 kg of urea because of the I said, if we charge uh, biochar with the urea, then they can substitute up to 51 kg of urea, 60 kg of MOP. That accounts for around Nepalese 4,400 rupees. That is also subsidized fertilizer. Otherwise, if carbon trading is legalized in agriculture in Nepal, we don't. It's not legal in agriculture sector, but it is legal in forestry sector. So if that is legalized, in that case, a farmer can earn more than 8,000 rupees per year with the use of biochar, only 1 ton biochar each year. So that should be legalized. This is what my recommendations and way forward. So this much, I thank you very much for listening me, and I also thank the chair and organizing committee. Thank you very much.
Thank you. Have a nice presentation. Again, of course, I have covered various beneficial aspects of the biochar in agriculture, crop management, and also crop production to increase the crop production. And when compared to even other inorganic fertilizers, it is proved that is how it's beneficial, and it has been proved beyond doubt that biochar is very useful in agriculture and also soil management. So thank you, Sri Prasad Mishra, for your excellent presentation. I think I will move on to next presentation is by Dr. B.K. Chakravarti. Before that, I would like to know any other speakers, particularly keynote speakers, are available here. Because one is Meena, is there? HP Meena. If he is, one second, Meena is here? No. So if there are no other listed speakers here, then I will take out two other presentation later. One Dr. Manjunath and then Dr. Rashmi. This under keynote presentations. Then later 30 oral presentation will start. 30. 10 minutes. So you please concise and conclude in 10 minutes. One second. Just introduce our speaker, Dr. B.K. Chakravarti. Full name, Vinay Kumar Chakravarti. Of course, he is a visiting professor, Swami Vivekananda Subharti University, and he is also Catadona State University, Philippines. Then is executive director and consultant of mud crab aquaculture management center, the supervisor Bangladesh Agriculture University, then former project director and Department of Fisheries, Government of Bangladesh. So he is a good friend. He has been associated in various conferences that we organize on behalf of the ATDA Society, and he is an extensive traveler. He travels all over the world and attend conferences. So I invite Dr. V.K. Chakravarti.
Thank you, Chairman. Thank you, dignitaries and academician. Good afternoon to all. Actually, my topics is on aquatic organism, aquatic environment related. I shall try to complete it within due time. Thank you to all again. Sorry. This is my. I am from Bangladesh. This is my Bangladesh. Bangladesh is bounded by India. Nothing to say because time is very limited. Now I want to say coastal area of Bangladesh. A land area 47,201 kilometers square. 30% of total land mass. Districts include 19 districts. Population is roughly 35 million. People, that is 29% of the total population is covered in coastal area. Ecosystem features such as Sundarban mangrove, forest, beach, and coastal reef, that is dams and wetlands. And geographical zone categorized into three regions, that are western region, this is Bonga, tidal plain characters and active deltic features with the numerous channel and creeks, and central region active area extending that is continuous erosion and acceleration, including Magna River century. Its zone is dominated by hilly terrain. Yeah, coastal water source in Bangladesh, it is if we categorize, then it is estuary, coastal line, backwaters, mangrove, lion, and aquaculture practice in the low-lying coastal plains districts of Satkhira, Pune, Bagarhat, with the southeastern Coxbazar region. About 1,093 aquatic marine organisms include finfish, shellfish, shrimps, seals, etc. Yeah. Marine, there is marine, that is area of where Bengal is 1,18,813 square kilometers, coastal line is 710 kilometer, and marine production is only 0.6812 million metric ton, and total production of Bangladesh was 5.018 million metric ton. And it is very much related to the seven SDG, that is 17 Sustainable Development Goal. Here it is very much linked with the 9 SDG, and SDG rank is 104, 104 out of 163. And I want to say one thing is that actually the most of us are alongside that is quoting the Orthosastro. Early mentioned in fish farming of agricultural tradition found in Kothulu Sastro, that is 321 to 300 BC, and King Sohestori, that is 1127 AD. And 1980, yeah. 1878, the renowned scientist of India, he wrote a book that is Fishes of India and Fauna of British India. So this is the background of ancient in fish culture. And here is another information about that discovered early method of fishing, harvesting, and aquatic resource management in the historic cities of Harappa and Mohenjo-daro. Harappa and Mohenjo-daro was the civilized area before first time. How they practice the capture of fish, the fishermen, and now methodology. This work is done by FGT, LEK, and directly with the individual respondent fisher farmer, that is transporter by another like this. Secondary data was collected from the mainly Department of Fisheries and literature online. Yeah, in coastal area, we are culturing that is another Asian sea bass, the latest skeleton here, and milkfish, and that is it is for cobia, and that is pearl spot species, and Mughal, the seven years, that is Grema bullet, and another is a grouper. These six kind of species are we are culturing in coastal area. And what is the production of our marine side? Here, there is the burning issue. In 1983-84, the production was only 0.16488 billion metric ton. It is increased. But last year, this dropped. So it is a very burning issue for our that is marine fisheries side or marine aquaculture. But the scenario of this, the decreasing contribution of marine fisheries production, 1984 the production was 21.88%. It decreased, and last year it is only 12.53% contributed to the total production. Why it is? It is due to climate change, and that is anthropogenic activities. That is overexploitation, filtration, that is indiscriminate use of pesticide, that is pollution like this. And yeah, this is the part of the total production, the traveling fishing. It is increasing. Last year it is decreased. This is called artisanal fish catch. It is also like this. Last year it is decreased. But production of river and estuary, it is very much connected with the coastal river. The production is increased. Why it is increased? In this region of one technology is Hilsa management program. We know that Hilsa is anadromous fish. It comes to freshwater for breeding purpose. And in Bangladesh, this is in Bangladesh. There is a technology only fisheries management technology is applied in the our open water. That is the coastal and big river, the Porda, Meghna, Jamuna, like this. For that reason, at that time there are so many species breed, and the spawn and coming generation is increased. Coming generation is what can I say? The population is increased for that reason. For that reason, the river production is increased, but last year it is decreased. So it is a burning issue. And what is happening about Sundarban mangrove area? It is a very important area and very sensitive area. The production is there increase. There are so many reasons for increasing the production, and 8 species are contributed to spawn culture. There are tiger shrimp, white shrimp, banana shrimp, white leg shrimp, and green tiger shrimp, emerald shrimp, and brown shrimp, yellow shrimp. These 8 species we are culturing. So
Dr. Chakravarti, please conclude in 1 minute. 1 minute.
The production of shrimp like this, it is increasing. And in the market culture, there are 4 species we are culturing. It is ongoing. Juvenile culture, crab culture, and crab fattening cases. And production of crab, it is also downward last year. So it is a burning issue. And the commercial fishing ground also, there are 4 fishing ground in Bay of Bengal. We are culturing. There are oyster culture in Bangladesh. There are so many species, 5, 6. That is 5 to 6 species that we are culturing. Here, cultivation of mussel is this is the culture method, and there are 5 species of the turtle. We are found in the Bay of Bengal. We are capturing. And seaweed now is the burning issue. It is very much related to burning. The seaweeds are culturing Kokbodar and Bakekali estuary, which southern coast and Andamanic river. What is the seaweeds culture practice in Bangladesh? Culture site is there is San Martin and Bakekali seaweeds. That is cultivated species is like 1, 2, 3, 4, 5, 6, 7, 8 species. So that is 10 species are culturing. That is red algae, brown algae, and green algae. There are so species included. There are these species we are culturing. Importance of seaweed. Seaweeds are very important physical colors. This is agar, alginic acid, and green is produced. Sea grass is a staple food also. You can seaweed salads. Medical uses purpose is seaweed is used. It is very important. Agricultural application it is very important. Rule of paper manufacturing is important. Important energy and bioactive component. Seaweed serve as a bioenergy source, so it is very important. Challenge of coastal aquaculture. There are so many challenges. Structural intervention is one challenge. Sedimentation is another challenge. And overexploitation, it is one challenge. Acidification is another challenge. So pollution is other challenge. Agrochemicals is very sensitive issue. there are 250 varieties of pesticides used in agriculture side. So contamination of soil and water enter into the food web and causes bioaccumulation of toxic substance and use 48,690 tons of pesticides yearly. So it is a very burning issue.
Go to the last slide.
Plastic and industrial effluent, oil pollution, radioactive pollution, transboundary pollution. There is a picture of how to this industrial domestic agricultural waste create pollution and what is the side effect of human being. And ghost fishing, human bias, commercial oiling and heavy destruction that is irresponsible fish farming also is a burning issue for aquatic ecosystem impact and effect of climate change. It is a very burning issue. I want to say there is a the climate change scenario in Sylhet region. This is the scenario, and this is the scenario of sheep culture. What is happening? 40 to 50% sheep already died for the climate change effect. And needs to measure to save coastal Sundarban for the there are so many measures are here and conclusion. To enlarge blue economy, focus on marine and coastal fisheries and marine biotechnology also, mariculture and the marine pollution, that is climate condition and marine, that is special planning should be required. Identifying causes of development of coastal fisheries, that is, what is that? Lack of implementation, enforcement, management of measure, limited planning and coordination to ensure quality seed and feed for aquaculture. And decrease the competition among the land, water, and feed user, promote low cost aquaculture and control pollution, need collaborative support between Department of Fisheries, between Department of Agriculture, Environment, Forest, Nodi Commission, Oceanography, and Land Department. Require for protection and conservation of coastal and marine fisheries resources are what needed, that is research, management strategies, and government policy for marine resources and implementation of acts and rules. And there are some references. This is the Thai seafood market I visited last May. We need this type of seafood production because Thailand is very rich in seafood production. So I want to conclude. Thank you to all.
Okay, thank you for your nice presentation. And Dr. D.K. Chakraborty covered most of the aspects related to coastal aquaculture. What are the commercially important species, fish species, then mollusk species, and other species culture. In fact, I am also coming from fisheries background. There is a talk by me actually listed here as a keynote speaker because of the time constraint. And in fact, I am skipping. I'm giving the opportunity to other speakers to speak. And okay, thank you, V.K. Chakraborty. Next, we move on to other presentations under the keynote speaker, Dr. Manjuna, Professor, University of Horticulture, Science, Bhagalpur. Yeah. So you're working at headquarters, Bhagalpur?
No, sir, I'm in the Mysore College of Horticulture.
Oh, horticulture. Over here, local.
Yes, sir, yes, sir.
I'm
the alumni of the Mysore University, sir.
Right.
Yes,
sir. Okay.
So, good afternoon.
You have about 8 minutes.
Yes, sir.
Yeah, please.
So, in the positive of time, I'll share my few of my work and working areas, and we are working with the farmers. So, topic of my presentation is a candidate bioagents for horticulture crop protection. Please next slide. Yeah. So, why we need the candidate bioagents? First of all, what is the meaning of that? So, candidate bioagents is nothing but a crop specific, a disease specific, a problem specific. It may be biotic or abiotic. Even the bioagents or the microbe we are employing for abiotic stress also now very specific. In such a context, I use the topic as the candidate bioagents. And recently, over the last years, that is very specifically studied. The pesticide, chemical pesticide usage is drastically reduced. Nearly 30% of the chemical pesticide is being reduced. And other data we have recorded from the farmers, nearly more than 500 farmers we are recording who are coming to us. And they are also using less than if they are using 100 rupees pesticide, now they are using 60 rupees pesticides. So that means the 40% of the pesticide expenditure is being reduced. That is because of the microbes usage, and not just microbe usage, and also they are aware of that. And what could be the best for horticulture crop protection, particularly the cash crops? For example, like ginger, pomegranate, grape, and so on. So this is what is the shift we are looking, and that's where the candidate bioagents come to the picture of the farmers. And for candidate bioagents usage and research, the most important the technology is like the researchers are using is the microbiome studies using the omics approach, particularly the metagenomics approach. What is that like the advantages? The advantage is the phytomicrobe studies by metagenomics and other microbiome studies. We able to understand the interactions between the host and pathogen, which able to identify the best beneficial the microbes, and also we can also identify the metabolites associated with the bioagents, which can be specifically we can apply to a particular problem. And this is the very schematic presentations of the phytomicrobiome studies in the metagenomics. If they are really interested, you can pick it up. And why phytomicrobiome studies is required for identifying the candidate microbes? Actually, by identifying generally, traditionally, over last 10 years back, only antagonist studies were used for identifying the biocontrol agents, and also some part studies and field experiments. Nowadays, after advent of the metagenomics approach, we are able to identify the gene level, what are the genes are present in the particular rhizosphere, and we can pick what genes you want. Suppose a particular plant has a 500 microbe, we can pick which is most efficient and which is very specific to the particular problem. For example, a pomegranate crop has the two diseases. One disease is the wilt and another disease is the blight. And we can identify what is the best for the blight and what is the best for the wilt. And that's how very precisely we can identify an organism, a candidate organism, by using the phytomicrobiome studies. That's why the functional characters, the functional attributes of the microbes are also can be identified using the phytomicrobiome studies. And this is like another the representation. Definitely, this phytomicrobiome studies using the microbiome, the omics approach, we can effectively identify the microbes, the candidate microbes. And the another important thing, what I like to share, actually the microbes are not simply colonized in the plants, and the plants metabolite actually attract what microbes they want. One very rare incident I like to share here. I'm working on the Nanjangud Raspberry. The Nanjangud Raspberry, one variety, the GI variety, is growing in the Nanjangud. Actually, we planted across the country over the 8 places. In 8 places, the 90% of the organisms are safe. Like only 10% of the organisms are different. That means the Nanjangud Raspberry are recruiting what microbes they want. That is the meaning. Actually, the metabolites of the plant is deciding what microbes they want. In such a case, definitely the candidate microbes will work, and it can be the future technology for managing the horticulture crop diseases. And again, this is what is the picture I wanted to share. I am sharing the Nanjangud Raspberry. Of course, the data I am not presenting, still it is under the progress. So I summarize, actually the microbiome, what I am telling, the candidate microbes are depending on the genotype and the seed microbiome, the root, even the architecture, the soil properties, and the soil qualities are also important. Again, I take the example of the Nanjangud Raspberry. Of course, I am working now. The Nanjangud Raspberry has a specific taste, which is very specific taste. Like the pulp is very specific in nature, and which cannot be identified in any other particular variety. Actually, that is attributed by the salinity of the soil. The salinity of the soil is attracting particular species like a Bacillus subtilis, which is only available in the saline soil of Nanjangud area, where it is also managing the wilt naturally. We are identifying by using approach of the phytomicrobiome studies using the metagenomics. And as I said, I'll summarize the physiology of the plant and the cultivar effect I'm trying. Only Nanjangud rust poly is attracting the Bacillus subtilis. If it is a plant of Cavendish is there, it is not attracting. That is what is the specificity and where the candidate microbes are having a role. And I'm taking another example where I'm working with the pomegranate. Actually, the wilt, actually pomegranate is being called as the disease crop. It is like it is a crop of the problem, and the one is the wilt. And we approach for the managing the wilt using a whole genome sequence and also metagenomics approach. And actually, the phytomicrobial studies also give, I said, also giving how the metabolite of the microbe is also contributing. We identified one compound like indole related compounds, the indole acid, like indole acetic acid, IAA compounds are also helps to reduce the pathogenicity of the pathogen in the wilt. Hence, we employed the integrated disease management after this study, the whole genome approach, where the boric acid can be applied in the integrated disease management approach. And this is the inventory of the microbes we have for the pomegranate. And we have like out of the 18 microbes, we commercialized 3 microbes. And this is the different studies, even for like the wilt is the complex of the root knot and other ceratocystis organisms. And this is the clearly says that the endophytes are very efficient, the microbes for the management of the wilt. And this is another study. And we commercialized actually out of 18 microbes, we identified and this is we commercialized the trichocavach. It is the combinations of the Trichoderma aspergillum, Pseudomonas bifida, and the Purpurosylium dilacinus. Combining those things, we develop and commercialize. And I am saying this is the candidate microbial consortia for the wilt management that is effectively using. I mean, the farmers are applying, and annually it is being sold by the government of Karnataka not less than 4 crores. This is what is our lab, the research work. Of course, it is like the consortium.
Please conclude.
Yes, sir. Yes, sir. So, of course, a few of the points I like to share. There is much data about that. And also, the volatiles produced by some of the bacterial endophytes after the colonization is contributing for the disease management. And they are only specific to the pomegranate that is able to manage the bacterial blight. So, the data I am not able to present here. And why the candidate microbes are required? Actually, the candidate microbes is not only giving the protection by inhibiting the pathogen, and also the metabolically it alters the host. So, for example, the salicylic acid pathway is changed and upregulated and triggered. And also, similarly, the japonic acid pathway is the triggered. I mean, in a different time point, and followed by the nitric oxide pathway is also triggered. That is what is the role of the candidate microbes for particularly the disease management. Of course, these are all the few of the publications. And also, I like to share that we identified the few microbes like the different approach. Suppose if the organism is growing and causing the epidemics, like from the 1 days to 7 days, the first day, the one microbe has a role, and the 7th day, the different organism has the role. So, out of the phytomicrobiome studies and the metagenomics approach, we can apply what is the first day to be applied, what is the 7th day to be applied. Where this kind of the knowledge, this kind of the awareness, we are generating the formulations to reduce the pesticides. This is what is required to make the biocontrol approach more sustainable and farmer friendly. Actually, I think this is what I wanted to say and conclude. Thank you, sir.
Thank you, Dr. Manjunath, for your excellent presentation. So we'll next move on to next presentation. This is the last keynote presentation by Dr. Rashmi. She is Associate Professor, University of Horticulture Sciences, Bagalkot. So you are also Bangalore, okay. She is presently working in Bangalore campus. After that, we will move on to oral presentations. Short oral presentation, serial number 1 to 30. Still, I understand some of the members have not uploaded your presentations on the system. So you please go and immediately upload it. Otherwise, you will not be able to present here.
Good afternoon, everyone. I'll be presenting today bioactive compounds from underutilized fruits and vegetables for their prospective entrepreneurship opportunities, especially in functional food and nutraceutical sector. So I'm representing UHS Bagalkot, which is first of its kind in specialized in our state, Horticulture University, as well as it's in third in the country. So we have almost 10 constituent colleges, and our mandate being teaching, research, and extension. We have a National Excellence Award from ICF from past 4 consecutive years. So with this, like underutilized crops, fruits and vegetables, like we have in tropical countries, which are very under-researched as well as less shelf life, and it doesn't enter the global supply chain. So we have to get into this, like segregating where we can fit in for the nutraceutical production. So these are the basics, like it might be traditional nutraceutical, non-traditional nutraceutical, that is fortified, as well as recombinant nutraceutical. So coming to the bioactive compounds, it's the secondary metabolites which are used for all the living organisms as well as cells, tissues, and then which imparts in distinguishing characters for color, aroma, as well as flavor to the fruit and vegetable. Accordingly, it is for overall health improvement. So with this, let me just brush off like what and all we have with that. Like it is classified as terpenoids, and then segregated with the vegetables in which it is highly evidential, and then flavonoid compounds. With that, we have gluconoids which are from brassica vegetables. With that, again, we have a miscellaneous, that is chlorophylls, amines, as well as creatinine and coenzyme and all these things. So in case if you want to go further with these bioactive compounds, this is the second one where we can extraction as well as using it in food function as well as cosmetic and industrial utility. So this is a set of flowchart where we can go. With that, we have identified which phenolic acids from which vegetables to which health benefits we are having it. With the one more thing to go for, when we are processing the fruits and vegetables, we have to look for how much it is retained. So in few of them, like it gets increased because of certain processing techniques. In few, there is no change, like soaking and overnight retention in water, whereas it decreases in peeling as well as conventional cooking methodology. So all these things we have to keep in mind. And then when we go further, for in case if you pick up a problem, like which problem we have to face with the bioactive compounds, I've taken up soil transmitted helminthiasis, in which it's a prevalent one in India as well as all over world. So especially for the small kids, it is more prevalent. So in case if you want to go for a nutraceutical anti-helminthic bioactive compound, we can pick up crops such as Suriname cherry, which is very good in bioactive compounds and nutritional composition. Along with that study, we could see that we have done the phytochemical constituents of Suriname cherry, which TPC, tannin content, as well as flavonoid content and alkaline content we assessed. We took up the different sectors of fruit and vegetable, and then we did a sequential solvent extraction, and we could come to the conclusion like mature pulp extract, that is ethanol extract, is the one which we got it. And then in that we have quantified the phenolic compounds, in which almost 14 different phenolic compounds were found. And then we had one more in that, again, isolation and characterization, where with that extract we could go, and then we could see that in one of the fraction we could identify so many compounds which, when we did an analysis, we could see that it was correlated to anti-elementic activity. With this, one more crop when we have studied, that is Momardica dioica, that is pine gourd, which is most prevalent in western Ghats of India. Here also we could find that the the quantification of phenolic compounds, we could make and it would correlate it with the anti-elementic activity. And then here, with the conclusion, we could see that caffeic acid, camphorol, apigenin, and TFC and TPC correlated for the antioxidant property, whereas caffeic acid, camphorol, and synergic acid and apigenin, synapic acid also correlated for the anti-elementic activity. with this, we have come out with one product, nutraceutical product, that is jelly gummies, that is with Suriname cherry and spine guard fruit paste, which we have taken up, and then doing all the initial work, we have concluded it. So one more thing is like how the leaves are more used, but we should know in that which constituent are more. So we did a study. Actually, this was sponsored by ICR in IDP project, and we did it in University of Birmingham. So where total ascorbic acid in different crops, as well as phytic acid, which is considered as a health one, as well as a it is anti-nutrient. There also we have to consider, because when you're consuming too much of phytic acid, it is not good, because it will restrict the absorption of minerals such as zinc, calcium, and iron. So we have to look for, and how can we combat the effect of phytic acid also should be done. So one more popular underutilized fruit, that is Singapore cherry, we have worked, which is a avenue tree where a lot of fruits goes as waste. In that, we have developed fruit squash as well as gummies. And then probably this is a canister, which is also called as egg fruit. In this, we have developed a energy-rich convenient snack, that is a fruit bar, with standardizing the protocol for the constituent. So along with that, in our college, we have an entrepreneurship development course in UG. So for 6 months, they go for, and they sell the brand in the name of Krishna Prabha NutriFresh with FSSA registration, so that learning while they earn while they learn. So with that, they do lot many products, and papaya leaf powder is also a very nutraceutical product where platelet count can be enhanced, and then usually for dengue. And then we develop lot many products like pseudostem candies, and then baby foods. And again, that and all, again, utilizing the prior knowledge of the nutraceutical value, and turmeric processing also, since at their UG level, they learn the processing and then selling it off also. And then cashew apple, which is nuts are used and apples are thrown. We do wines out of it. And again, we promote marketing and sales for the students, so that they learn the entrepreneurship. So with this, we
Please,
have an outreach program.
thank you.
So this is the one which I want to conclude. That is science knowing no country, because knowledge belongs to all. And this is a torch which illuminates the world. With this, let me conclude my talk. Thank you for the patient hearing.
Thank you very much for your nice presentation. And of course, this concludes all the keynote addresses and keynote presentations. Then we move on to next for oral short presentations. And I think this third session is going to start. In this session, is chaired by Dr. Rajkumar H. Karampalli, the chairman of studies in botany, Mysore University, and chair of rural okay. Then co-chaired by Dr. Manjunath. Manjunath is here? Yeah, so okay. So I think 30 presentations will be there, and 5 minutes will be given for each presentation. So try to be crisp and don't go for any introduction, that, this, like that. There's no, otherwise you don't get time for your final results. So straight away go to material methods and results. Okay. So I think I'll hand over. Next class.
Okay, good afternoon one and all. So I welcome you all to this oral presentation. As sir mentioned, the time allotted is 5 to 10 minutes. I think 5 to 8 minutes it has been reduced. So I request you to stick on to the schedule and present your gist of your work. Okay. So first one is Dr. Mahadeva J.
So good afternoon to everybody. So I am going to present the paper, a part of research paper. So how the potting mixtures are potting media and germination seedling growth in Biodiversity. Actually, why you select the Biodiversity is totally it's one of the friendly the tree for the farmers. And then it's a multi-purpose trees. It's used for the various purpose like even the fodder and the green manures and also pulp and paper industries and also the use for the furnitures and any other things. It is increase the rest the farmer the income. So whenever the growing of even the media duvia, it's commonly called as the neem trees. So in Kannada, it was Eppeu. This is one of the important trees, fast growing and multi-purpose the trees. Thank you. So it's a potential purpose trees, ideal agroforestry, increase income per unit area for the water. Actually, this is the trees very useful for the farmers. So it's a growing fast growing trees. So and it's a money making trees for the farmers. Rotation, strong demand for plywood, lumber industries, and also the furnitures. Also the use for the fodder and sometimes used for even the manure also. It's a high yielding the fuel and fodder. Also, it's a wide range of medicinal, pharmacological, and traditional characteristics. Many other uses. So the fast growing forest tree species which has been multi-purpose uses. So long time germinator which may vary from too much of 2 years. Its germination percentage very low, which is around 25% only. The number of studies are conducted in germination of the leaves are still lacking information because of why we select this the trees. It is essential for the farmers for the purpose of the agroforestry. So the details of proper treatment methods or standard germination protocol is very required. So the studies conducted with the interest of finding different methods of technology, given the initial germination percentage, because it's very difficult to germinate the seed because the seed the core is contain more the high hard content. That's why it's very difficult to so germinate. So that's why I selected these topics. So what are the objectives? The small the area actually to study the effect of different potting media and seed germination and the media duvia. Study the effect of different potting media, seeding growth and media duvia. So some of the experiments were conducted in a very speed, particularly selected in the TKVK, the thesis media duvia and take up experiment in laboratory conditions. A treatment taken the 9 and application is 4. Expert designs and CRDs and treatment details. So there is a 9 treatment the details. So control and soaking with sulfur water at 1% for 20 minutes and T3. So treatment 3 is scarification with sulfuric acid and 1% for 20 minutes and T4 soaking with potassium nitrate 1% for 20 minutes and treatment with liberal acid at 500 ppm for 6 hours. The treatment with cytokines at 100 ppm for 8 hours and treatment with naphthalene acetic acid so at 100 ppm for 6 hours and are soaked with cold blood slurries for 10 days and dried for 2 days and repeated 3 days soaking with the water for 6 hours. So these are the experiments were conducted in different in the laboratory conditions. So these are the result for the batch. So totally germination of the and seeds, other treatment, the different treatments. So totally the germination seeds. So T3 is actually is a source the highest germination when compared to others. So and some of the seeds, normal seeds about 18 43 1.5 abnormal. So these are the result were taken by the some of the different methods. So germination seeds after seed treatment with sulfuric acid treated with the powder. These are the so the good result when compared to other treatment. So seed germination percentage of million gas. So totally that's when compared to T3, so the highest the values, highest germinations. So compared to the treatment process. So and among the selected treatment, T3 is scarification with sulfuric acid and 1% So the highest germination 63.88% because of high acidity which corrode the seed of coat and helps in speeding of the germination. And second highest germination obtained in the T8, so seed soaked with the codex, so they have 10 days and dried to 2 days and repeated 3 times, and it is 54.05% and will decrease hardness of the seed coats. And the lowest is the T1, so which is so 26.8%. This may be due to the presence of large quantity of hard seeds. Second lowest of germination seed in the production related, which is 28.7% due to neutral pH levels. So totally unable to loosen the seed course. So T2, so soaking with waters after 1% of the 20 minutes. Also, so they are the minute increase in germination percentage with 30.88% because of its weak acidity, the H+. So finally, T5, T6, and T7, which are the growth regulating hormones, did not show that much of the difference of germinations because the 34.2, 32.5% because of because of the hormone cannot exert because of the potential due to the extreme hardness. So finally, what we are going to conclude, the seed germination percentage in the media seeds are low due to the presence of hard seeds, dormant seeds, and large proportions along with the minimum portion of the dead seeds. However, these issues can be solved by the subjecting seeds for the acidic application, treating with the powders, treatment for the sulfuric acid source. This varies. Germination treatment with for the dead, so the 2% germination. Thank you.
If there are any one or two questions, the audience may ask. Okay, if there are no questions, so among the treatment, in that T3.
T3 sulfuric acid and followed by codex.
So how the farmers have to follow actually?
So actually,
Because you use H2S.
already I suggest the farmers. The farmers are using that method also because the media seeds are very the hearts the seeds are very difficult to even the germinating the seeds.
No, you used for scarification plus this H2S4.
H2S4.
They know how to use H2S4 and all.
So actually, we have implemented this the methods. The farmer already trained up the farmers some places. So, but no one to come in our places. But farmers agree with the follow these methods.
Okay, thank you.
Suchi Mehra. I think he's not. Amit Tavana. Krishnalal. What's the name?
Krishnalal. Second number.
It's not here. Here is a other one. Krishnalal. Yes, sir. Yes, sir. Yes, sir.
Respected chairpersons and dear delegates, good afternoon to you all. The topic of my presentation is current status of fish catch from Pond Reservoir and Govind Sagar Reservoir, Himachal Pradesh. Pond Reservoir has been created by damming river Bias in 1975, and Govind Sagar Reservoir has been created in 1963 by damming Satluj River. Both these reservoirs are large reservoirs and located in Himachal Pradesh, and this data is concerned with 2023 and 24 years. And different conservation measures are being practiced in Himachal Pradesh. And for 2 months, there is no fishing. So these months are starting from mid-June to mid-August. And these are for conservation reason, and during these months fish breed, most of the fish breed. Cyprinus carpio, that is common carp, is also being stocked regularly along with Indian major carps in Himachal Pradesh, these reservoirs. And Hypothalamitis molatex, that is silver carp, have got accidentally introduced in Govind Sagar Reservoir. Strata Singhala was 40.2 tons. All this data is in tons. And in one of the reservoirs, that is in Govind Sagar Reservoir, and it was 182.75 tons in Pond Reservoir. So Pond Reservoir is being dominated with the catch of Spirata Singhala. Tinopharagodon idella, that is grass carp, is very less in these reservoirs because aquatic weeds are not available in these reservoirs. So contribution of silver carp is highest in Govind Sagar Reservoir. Sprata Sinhala is highest in Pong Reservoir. Only licensed fishermen are allowed to do fishing in these reservoirs, and they have organized themselves into cooperative societies. And other fish in these reservoirs are Katragata, Labiorohita, Tor putitora, that is golden mahashel. This is a state fish of Himachal Pradesh. Besides it, Levio, Daru, Levio, Calvasus, Rihana Brigala, Channa, Marulius, Sprinus, Cyprinus, Carpio, etc. are also there. So this is the catch composition of this year. And just after the close season, that is during the September month, most of the catch is there. So this close season has a positive impact on the production of the fish. And minimum catch was during the winter season. And maximum catch in this case in Govind Sagar Reservoir is belonging to silver carp, that is Hypothermicus molitrix. And on second position is common carp. In Pond Reservoir, on first position is Pirata Sinhala, and besides it, Levia Rohita is also high in catch. 45 tons catch was there in year 2023 to 24. This was belonging to Levia Rohita, and Torpitutora was also much in this catch. Leviodero was not there in Pond Reservoir. This fish, these organized societies started working in Govind Sagar Reservoir in 1960s, 1964. With 16 fishermen started their work, and silt deposition is also getting there in these reservoirs, and hindrance is there for breeding of the fish. Above the Govind Sagar reservoirs, one more reservoir was constructed, that is Coal Dam Reservoir, and due to that dam, fish migration got hindered, and adversely there was impact. Adverse impact was there on the fish due to that dam. So maximum catch in Govind Sagar Reservoir was belonging to silver carp, which got accidentally introduced into this reservoir in 1971. And besides it, Cyprinus carpi was high in catch, Balago Atu, and the Channa species were not in Govind Sagar Reservoir, but these were in Pong Reservoir. In Pong Reservoir, highest catch of Sprata Sinhala was there, but silver carp, that is Hypothermicus moltrix, was less in catch. Grass carp was not in Pong Reservoir.
Okay, so you presented well in time, so that's really good. And what would be the reason for variation of catches in different reservoirs?
Sir, water temperature.
Temperature,
Yeah.
that is the only reason.
No, other reasons are also there, and these catch efforts are also because these are big reservoirs, and so much these parameters are not being studied. So sometimes more fishermen are there, sometimes less fishermen are working there, and water fluctuations are also there. In one of the reservoirs, water fluctuation is more in Govind Sagar Reservoir.
For conducting research, you must standardize something for calculating the catch and the numbers, everything.
Yes.
That's what I understood.
Yes.
So what could be the reason actually?
So different reasons are there. So temperature.
You are mentioning
One
temperature
is
is the only.
pardon?
Only temperature.
Temperature, and then these gill nets are being applied, and minimum size of the that gill net should be 5 centimeter, more than that. But variation in those gill nets is there. Some people are using this gill net having mesh size of 4 inches. Some are having more, less. This type things are there. Uniformity is not there.
Okay, thank you.
Thanks.
Amitabh.
The next, I think there are no Pooja Tiwari, Jean Mary, Atman Punia here. Okay. So next presenter is Dara Nishit Zaveri. Okay, fine.
Good afternoon, everyone. My topic of presentation is a novel approach of using ash gerd as a substrate for enhancing lignocellulolytic enzyme production in mono and co-culture of two white rot fungi. Here by this, we all know that white rot fungi produces many lignocellulolytic enzymes, which are actually having many biotechnological, environmental, and industrial applications. And to enhance that, there is two techniques supplied here in this paper. One of the technique is co-culture technique, wherein two fungi grows together, and another technique is to provide some suitable substrate for growth. Here in first, we have selected these two fungi, Ipex and Dedelioxys conflagrosa, in monoculture also and in co-culture also. And as a substrate, we have selected this ash gourd, which is prepared differently. So here, in materials and method, we have collected the Ipex and Dedelioxys conflagrosa culture from FRI Dehradun, and then we have performed the parent interaction test to know the compatibility of both. Then we have used ash gourd as a substrate, and here in ash gourd, we have peeled and seeds of the ash gourd were removed, and small pieces of the ash gourd were dried at 80 degrees centigrade, and thus prepared substrate which will be used for the further experiments. Here, for experimental setup, I have taken 3% mal extract medium, and in that, after sterilization, for monoculture, added one disc of 9 mm size, and for co-culture, one disc of both the fungi were added together. And for substrate substitution, percentage from 1 to 10% were taken, and in that, mono and co-culture both have performed. After this, we have taken out the filtrate crude extract of this, and that crude extract will be utilized for performing this enzyme assays. So we have performed MLP, LIP, lactase, AO, xylanase, and cellulase enzyme to check whether this production is enhanced or not. This is the results of the co-culture taste. Here we have grown the apex, and here we have grown the Dedelioxys conflagrosa. And after ninth day, this is the photograph of ninth day. This is where this both have overlapped, and this is grown further for 3 weeks. And growth for both the fungi were not inhibiting each other; they both were mutually intermingling. So they are not inhibiting the growth. So they have been selected for the further experimentation. Now this is the result for the enzyme production for monoculture, co-culture, and normally we in this experimental setup we have seen we have seen the effect of co-culture condition where the enzyme is secreted. So wherever in this lignin peroxidase test we have seen that it packs is not producing the LIP, but in monoculture condition and in co-culture condition, almost similar amount of enzyme is seen. In manganese peroxidase, here in co-culture, the highest amount of enzyme secretion we have got. Lactase is enhanced tremendously by using the co-culture condition. So wherein this light blue color graph is showing that the highest amount of lactase is seen there. AO and xylanase also showed the highest amount of the production by in the co-culture condition. In this all, we have seen that the highest amount of the enzyme produced were at the lower incubation days also. So that is also beneficial for the industries, that in less amount of incubation period you can get more amount of enzymes. Here in both these fungi, they do not produce cellulase enzyme, and they even in co-culture condition do not produce cellulase enzyme. After that, we have added the ash gourd pulp in different concentration. So this is controlled, and this is the different concentration of ash gourd. So here we have seen that in all these enzymes here Ipex does not produce LIP, and which does not produce even with the effect with the ash gourd also. But here we can see in comparison to control, all have a little bit higher amount of enzyme activity, and which is highest at 5%. In MMP also, in comparison to control, all of them and were having the highest enzyme activity. And in lacase also, the enzyme activity was the highest in 5% of sugar pulp. And then in A also and in xylamase also. Here also with the using by using ash guard, we could not find out the cellulase enzyme. Here we can also see that the enzyme activity which we were discussing, it is highest in case of co-culture condition and with the help of 5% xylanase. So 5% xylanase is the best enhancer which is proved here, and co-culture condition is also enhancing the enzyme activity which is beneficial to the industries. This is my references. Thank you.
What meant like if co-culture, it is enzyme activity be more?
Yes.
I suppose if you co-culture, I mean, with the using ash cloud,
Ash guard.
the enzymatic activity be more?
Remove, no, sorry, it is
Will
enhanced.
be more?
Will be more, yes, yes.
Okay, okay. So why you choose only ash card? Why not other cards?
Actually, Ashgard, there were some references. Ashgard is having some xylanase in that, xylanase and pectin, which is there in the Ashgard, which will be used as an enhancers for these enzymes. So that way we have gone. The enhancers for this is the xylanase and pectin, and then it is there in the Ashgard, and then we have selected the Ashgard as a natural source of both.
Thank you. Thank you. Dara? The next presenter is Matoshi Mandi. Is here? Yes, okay.
So good evening, chairpersons, respected dignitaries and all. My name is Dr. Dara Zuberi. I'm going to present my on the topic of the effect of salicylic acid on the morphological and biochemical parameters of Withania somnifera. Withania somnifera, basically a plant belongs to the Solanaceae and it has a lots of medicinal value, or it's also known commonly as ashwagandha. And in Ayurveda, it's known as ashwagandha and commonly known as the Indian ginseng. And it has a medicinal properties like the adaptogenic, anti-cancerous, immunocholinergic, and it has some of the neurological disorders. It can be used. It also can be used in arthritis and other disease also. And mainly in some of the Ayurveda, it mentioned that it's also used as a sedative. And because of that, I choose this plant for to enhance the phytochemical constituents which are there in the plant. And for that, I use enhancers, which is the salicylic acid. Salicylic acid is a natural plant growth hormone, and it has a lots of benefits. It can help the plant for the growth and development. It's also response. It helps the plant against any of the biotic and abiotic stress. It also can regulate the plant growth, and it also increase the activity of the photosynthetic enzymes and enhance the plant tolerance. Specifically, acid, that is salicylic acids, it affects also affects on the another plant growth hormones like the auxin, gibberellin, cytokinin, and other modulators. And some of the genes which are involved in the production of the salicylic acids that also help to trigger the other plant growth hormones. And these are the few of the basic biological function of the salicylic acid. It's help into the seed germination, leaf senescence, fruit yield. It's also help the plant against the stress condition like the drought condition, stress condition, and it help into the stomatal open and closing functioning. So these are the material methods which I followed. First, we collect the seeds from one of the gene bank, the CG gene bank we collected, and we just give the treatment of the salicylic acid with a different concentration, and then we just prepare the pots. In that, just we put that seeds, and then we measure the morphological measurements like the root length, shoot length, leaf numbers, biomass, and the leaf area. Biochemical estimations like the chlorophyll, proline, and the phenol also done for that. And stomatal index and the trichome index is also done as a part of the experiment. So these are the different parameters that we observed during the experiment, and the result is this: the leaf area, number of the leaves will be we are getting more in these the plant which is collected after the 9th weeks under the concentration of the 1 millimolar concentration. Shoot length also we are getting more after the we are getting harvested plant after the 9th week and in the 1.0 concentration. Root length we are getting more in the 0.5 molar milli concentration again collected after the 9th week, and the biomass also we are getting with respect to the 0.0 millimolar concentration. These are the other parameters like the leaf area. Leaf area also we are getting at the concentration of 0.5 millimolar after the 7th week. Chlorophyll estimation we did chlorophyll A, chlorophyll B, and the total chlorophyll estimation. That also we are getting in the 1.0 millimolar concentration. Amount of the phenol and the amount of the proline, we are also this amount of the phenol which we are getting in the 0.25 millimolar concentration, and the proline we are just observe under the 1 millimolar concentration. But when we keep this experiment for the longer time beyond the 9 to 10 weeks, so we are getting the good result in the 0.5 millimolar concentration. 1 millimolar concentration plant shows the degradation of the other secondary metabolites. These are the few representative photographs of the experiment which is set up. This plant under the control condition, this with the 0.25, this is 0.5 and the 1 millimolar concentration. This plant harvested after the 5 week, this one is after the 7th week, and this one is after the 9th week. These are the seedlings which are just taking out from the pots. This one under the control condition, this one is 0.25, 0.5 and the 1 millimolar concentration. Again, after harvested after the 5th week, 7th week and the 9th week. So when we plotted the plants, these are the life cycle which is going to be there. We are just germinate the seeds, and at the last we are getting the seed development also. These are the different, there is a type of the stomata we are getting. We are getting the anemocytic type of the stomata and the multicellular and the cellular trichomes we are getting in that. And this is the result that we are getting higher stomatal index in the 9th week of the 0.45 millimolar concentration. So these are the conclusion that the 0.5 millimolar concentration shows the high amount of the this chlorophyll content and other biochemical concentration as well as the stomatal index also high in that. So it is a best suitable enhancer in agriculture and the pharmaceutical for the medicinal plants. These are the references. Thank you.
Okay, so you mean to say that salicylic acid enhanced the growth aerial part of the plant? Normally, this ashwagandha, its root is used, no? Normally, useful part in ashwagandha.
Basically, whole plant is used, but different what purpose you are going to be used. That is also very important.
No, in Ayurveda,
Yeah.
root is used normally.
No, so leaves are also used.
Is it?
Yes, definitely.
Okay, so if the leaf is used, what is, you could have selected, tested the secondary metabolite ashwagandha which is there in that particular active compound,
In leaf.
which is, that's what, in leaf or it may be in root actually.
Sir,
So
basically-
that would have been added, that suggestion is that would have been added your work rather than going for all other parameters. Because medicinally, the pharmaceutical companies or the Ayurvedic company people, they look for its active principle.
Yes, definitely.
Okay, all the best.
Thank you.
So Matoshi, the next speaker is Dayamani KJ.
Hello, good afternoon, dignitaries and all other participants. I am Dr. Mokshi Mandi, and today in this wonderful platform, I am going to present a piece of my work which entitles protective interplay of vitamin C and E against acid-mediated reproductive disruption in Drosophila melanogaster. So this work has been conducted at Department of Zoology, the University of Bardhaman, and this Bardhaman district is considered as the major agricultural hub of West Bengal. And for this reason, it has owned the title as the granary of West Bengal. So to maintain the crops and to protect them from different types of pests, different types of pesticides are in use. So here comes my pest chemical, that is acephate, which is chemically organophosphate in nature. It mainly works as an acetylcholinesterase inhibitor. And here comes two commonly used dietary antioxidants, that is vitamin C and vitamin E, which we get from different types of natural food sources. And here comes the model organism, which is Drosophila melanogaster, which is an ideal model organism and a well-established model organism in the field of biological sciences. So in this study, Drosophila melanogaster is used as a non-target model organism. So the objectives of these studies are to explore the impacts of organophosphate pesticide acephate on particularly the reproductive health of Drosophila melanogaster, and moreover, this study also aims to assess antioxidant potential of two dietary antioxidants, that is vitamin C and vitamin E. So this is a brief scheme of the treatment schedule where first instal larvae were introduced to different treatment categories and adult male and female flies were isolated and taken for the experiments. This is the materials and methods and result portion. So as I am focusing on reproductive health of Drosophila melanogaster, so fecundity and fertility, that is represented as egg hatchability, are two important parameters. So this is the first result of fecundity, where after chronic sublethal exposure of acephate, a significant decrease in fecundity, that is calculated from the number of eggs laid per female individuals, is significantly lower, whereas after supplementation of vitamin C and vitamin E, the fecundity value is coming towards the normal value. The result of egg hatchability percentage is also similar type, that is after acephate exposure, the egg hatchability percentage is significantly decreased in compared to the control categories, and after supplementation with antioxidants, the hatchability percentage again restored towards the control level. Next, this is the pictorial representation of the reproductive tissues, that is for ovary and control, and these blue patches are representing tissue damage in the gonadal tissue. So much or deep blue patches are indicating higher degree of tissue damage, which is found in the acephate exposed category, whereas in control and in antioxidant supplemented category, lower number or lower intensity of blue patches are observed. This is one biochemical estimation, that is vitellogenin assay. So vitellogenin is the major yolk production protein in the egg-laying individuals. Vitellogenin assay also demonstrates significantly lower level after acephate exposure. And also this is a biochemical estimation of NAD, that is NAD-dependent malate dehydrogenase enzyme assay, which is an important enzyme, works in the TCA cycle, and this TCA cycle is the major enzyme or major biochemical pathways for production of different types of biological macromolecules, including amino acids and fatty acids. And these two biochemical estimations, that is mitochondrial assay, and after that the angiotensin converting enzyme. These two enzymes are two major enzymes in the male reproductive system, and both these angiotensin converting enzyme and mitochondrial plays crucial role in the spermatogenesis of male Drosophila melanogaster. Both these enzymes were significantly found lower activation after following the chronic exposure to acephate, and after supplementation of antioxidants, these activity are quite increased. And these are two biochemical parameters, that is MDA and lipid peroxidation. NDA, that means lipid peroxidation and protein carbonylation. Both these are directly and parameter of oxidative stress, indicator of oxidative stress. So after acephate exposure, both these oxidative stress indicating enzymes are indicating higher level. But after supplementation with the antioxidants, these oxidative stress indicating enzymes are found significantly lower. This is the pictorial representation of gonadal tissue where it is indicating the nuclear fragmentation in fluorescence exposure after that is staining. So a significant nuclear fragmentation were observed in the acephate exposed categories. This is the photographic representation of cometation and this is a very prominent parameter of genotoxic effect. So significant genotoxic effect is found after acephate exposure and significant restoration is also found after antioxidant supplementation. Yes. So these are the take-home messages. Chronic sublethal acephate exposure results in reproductive insufficiency and protective advantage of co-administration of the dietary antioxidants, vitamin C and E, validated the reproductive toxicity due to oxidative stress, protected from this oxidative stress. These are some selected references. And thank you for your patience hearing.
So acephate is being widely used in your spray state?
Yes, sir.
That's which state it is planned?
Yes, sir. But in my state, it is considered uses.
Exposing the acephate, if you eat vitamin C and vitamin E, we can overcome it.
No, actually not completely, but it is showing somewhat protective efficacy against these type of pesticide toxicity.
Okay.
Thank you.
Yamani, the next speaker is Chana Basava. Is here? Okay.
Good evening to the chairpersons, dignitaries, and the fellow delegates. Today I am going to present on my research that is conducted in COH College of Horticulture, Mysuru, under VHS Balcourt. It's on diversity analysis of the soil actinomycetes and their antagonistic characterization against the fungal plant pathogens. So nowadays we have seen that many of the pesticides are being applied to the soil and many crops against there's having the deleterious effect on all the group of microbes, including our soil actinomycetes. Soil actinomycetes are playing very good important role. They are playing very good important role in decomposition of the organic matter and also being the major producers of many of the antibiotics that today we are all using in both the medical purpose and also in application of agriculture field. So in this aspect, in this background, we have started exploring the collection of the many soil samples from the different horticulture crops, that is, and from the rhizospheres of banana, ginger, coconut, and from the soils of southern parts of Karnataka, that is in Dakshina Kannada, Chikmagalur, Hassan, and also a few parts of Mysore we have covered. And you can see the color variation in the soil samples we have collected, and they are subjected to isolation by using the many different types of media, and also the very well confirmed isolates through morphological variations, and they have taken to further biochemical characterization and also molecular characterization. You can see in this picture that is having the inhibition effect over the neighboring fungal cultures. And about 100 actinomycetes cultures were collected, and also they have again proved by microscopic observation and also by chemical characterization, and finally the molecular characterization was also done to confirm them. And also you can see the morphological characters and the difference between the different types of isolates or the strains. And also nowadays you can see the pigment variation among the collected isolates of the actinomycetes cultures. The pigments are being very widely used in the various industries, including the cosmetic, textiles, the dyeing industries, food industries, automotive, and also the paints and coatings and plastics. We can also move towards taking these actinomycete strains apart from using it as a good organic matter decomposer. As we are isolating, majority of the scientists, they used to isolate these actinomycetes
mainly from the compost, so that you can imagine the decomposition capacity of these actinomycetes strains. So now imagine they can also taken to the painting industries or also the pigment producing industries. And also, some of the students they're also working on this commercialization of the pigment production. You can see the variation. This is the microscopic observation of the actinomycetes strains. Many of the microbiologists may have observed these actinomycetes. They're somewhere in between the bacteria and fungi. Bacteria because they are gram positive, and also morphologically they looks like the fungi because they do produce the short mycelial-like structures. That why beginners, the microbiological beginners, they can somewhere they get confused about whether they're bacteria or the fungi. So again, they were tested for their antifungal activity, and also the secondary metabolites were also being extracted through chloroform and ethyl acetate combination. Again, they were checked against this is the fusarium culture. Many of the fusarium cultures they have inhibited up to 70%, 70 to some isolates. They have we have obtained up to 70%, 70 to 80% inhibition we have observed. You can see the colony inhibition. This is these actinomycete isolation, apart from having the producing the secondary metabolites, they can also be used as such. They are using in mainly the agriculture. So some of the commercialized products are being obtained. So like Spinosad, that is the very well recognized and also registered company that is being used in the advanced formulations of these actinomycete strains. And further, these actinomycete strains which are showing the potential inhibition activity, they were sent for the PCR amplicons were sent for the sequencing purpose to see whether any variation or the very diversified difference is there among the collection. But 95% homology lies between only the Streptomycetes species, as these Streptomycetes species are the cultures are dominating all the in the Actinomycetes group, followed by the Nocardia and Micromonospora. About 60 to 70% of the Actinomycetes is covered with only the Streptomyces species. That has been again proved with this type of experiments. Final conclusion is again that these Actinomycetes cultures or the strains can definitely be used in the organic farming or in the any pigment production industries. Definitely, there is no doubt that this can be explored very well. Thank you.
In almost all soil, the dominant species was Streptomyces.
Streptomyces.
So even in all the areas, regardless of any soil.
Sir, this is actually Dr. Manjunath sir is better know that was having the DST M scientist project. This is the extended work of that project. Though that DST M scientist project, we have collected the soil from all over the all parts of the India. So because our students and fellow colleagues, whenever they go for the trainings or conference, I used to collect through the soils from them. Almost we have seen that only the Streptomyces mycetes are dominating.
Because the physical chemical parameters of different conditions, soil condition varies.
Yes, sir. Because they are the dominant producers of the antibiotics. Many different sequential array of antibiotics, secondary metabolites are being produced.
Okay.
Thank you, sir.
Speaker, please. Channa Basava. And after that, Nanay sir. Yes, sir.
Good evening, one and all who have gathered here, respected dignitaries on the chair, and all the delegates who are witnessing the successful of this 12 witnessing this international conference at Mysore. Okay, good evening, all the delegates and respected dignitaries on the chair. Myself, Dr. Chanbaswa, would like to present my talk on mycorrhization effect on growth and acclimatization of micropropagated Aspergillus claviculotum, that is podomelid. As we are aware of the micropropagation is the method where we can develop a plant without infections or disease-free plants we can generate. And it is possible to develop a desired character through the micropropagation. That intention, the micropropagation method has been adapted. And the secondly, the coda millet is selected because nowadays we are facing the problems with respect to many health risks, and this millet is endangering. Now it is going away from the agriculture field. To promote the usage as well as the cultivation of this coda millet, I have selected this plant for the experimental purpose. And as you need to remember, this is a very much it is a long crop. We can store the seeds of this plant for 100 years because I have collected the seeds which are kept for more than 35 years from the one of the agriculturists in Koppal district. And from him I have collected, and those seeds are not germinated in the normal conditions and then tried with a micro propagation. And it is having very good dietary fibers, and it is having minerals very rich with respect to particularly iron, calcium, magnesium, etc. And material methods, as I said, the seeds were collected from the farmer and they are surface sterilized with a standard methods. And for the micropropagation, the basal media, MS media was used, and the plants were transplanted. And before doing this transplantation process, the acclimatization process, I have selected the two important mycorrhizal fungi, that is Rhizophagus fasciculatus as well as Glomus macrocarpum. And these two species were selected based on the biodiversity assessment of the 6th district in North Karnataka. And in all the North Karnataka, millet growing areas were represented by or dominated by these two species. Since I have selected these two species and marked multiplication of these two fungal species, mycorrhizal species were made by the trap culture method. And for the estimation, I have selected the parameters of the biochemical aspects like the protein as well as the chlorophyll, and all the values were subjected for statistical analysis by using ANOVA, and the comparative account was done with a DMRT, and Pearson correlation was analyzed for all the characteristic features. And this is the picture where we were going to observe with respect to, this is the method where how to isolate the spores, and this is how we have counted the spores, which are standard methods described by the Phillips and Iman and Jordan Nicholson. And these species were identified with the well-known manuals like comparison and recent advancement in the Glomeromycota trends. And these are the results. When I scanned for the roots of the inoculated plants, here I am able to observe the plant which is inoculated with Rhizophagus fasciculatus. I showed the more number of arbuscles as well as the vesicles. And this is the slide which we are going to consider here. The previous one. Next. This is the slide which we are going to consider. This is the plants which have grown under laboratory condition for the germination of the plantlets, and the same plantlets were initially transplanted to the plastic tub. Later it is transplanted to the earthen pots. Later the parameters were, that is root length, dry weight, and fresh weights of the vegetative parameters were measured. Accordingly, it was very good notable with respect to Rhizophagus fasciculatus. All the parameters with respect to length, fresh weight, and dry weight with respect to shoot and root were enhanced, but there is variation with respect to the mycorrhizal spore number. That is, MSN is nothing but mycorrhizal spore number. It was very good with respect to Glomus macrocarpum, but the percent colonization was good with respect to Rhizophagus fasciculatus. We need to remember the colonization impacts the growth as well as absorption of phosphate and other mineral nutrients which are essentially required for the growth of a plant. As the micro-propagated plants are very weak in nature and they are having the less conductive properties at initial stages, to enhance that mechanism, we have inoculated the mycorrhizal species, as these two AM fungus species are native to the areas where the millets are growing. And at the two different stages we have harvested, that is one at 60 and other at 30 days, and the PSN correlations. And as we need to remember, this micropropagation along with the mycorrhization enhances the stability and the hardness of the micropropagated plants, and it is easy for the transplantation, and the survival rate of the mycorrhization plants were maximum when we compare with respect to control plants. These are the references which I have referred. Thank you. We should think green and go green.
So how did you deliver? What is the mechanism? Did you formulate it and deliver for the field? Only it is the in vitro propagation experiment.
Sir, it was the seeds were very late now. They're away from the agriculture field, and the seeds collected from the farmer, they are not germinated in the normal conditions. Then I tried with micro propagation, and then I think of acclimatization effect with respect to mycorrhiza. That is one. And later I collected the seeds from the other farmer in Haveri, and they are grown naturally in the field. And at the same time, this experiment is collaborated with that field experiment also. The same result, hardening process and growth, root length, chlorophyll, and even at the last final stage of the harvesting seeds, the number of seeds were more per head of the plant was reported with respect to Rhizophagus fasciculatus when compared with the macrocarpum as well as the control plant. Thank you.
So, Nanesh sir, the next speaker is Deepak.
So good evening to all. Respected Chair, I request 5-6 minutes extra because this is a very important topic where morning Professor Niranjana sir was talking about artificial intelligence, machine learning, and precision farming, which is the need of the hour. So quickly, if you glance that 53 billion expected the exports, and we are already surpassed post-revolution, and currently we are focusing on the sustainability, where we have already achieved quality and quantity, but now the every talk is on sustainability. Out of the 17 goals, agriculture possesses 6 important goals to be covered. And what are the major challenges? And the best major challenges in India currently we are facing is increasing the population. As pointed in the morning, by 2050 we'll be reaching around 10 billion population. So currently in India we are already 1.4. And apart from that, the global average temperature, if you see, it's already 1.6. If it reaches to 2, we cannot produce much good foods as we expected, because most of the crops are sensitive to global temperature. And you can see the beautiful picture of the glacier melting, where the water level will keep on increasing. And over the years, you can see the first picture where the husband and wife went to honeymoon 15 years back. And when they come back to the same place, they cannot find any glacier. It's very important to note that. And also, you can see the Greenland glaciers in the USA, where completely degradation of the glaciers we can see. And other thing important is water stress in India, especially if you pinpoint here, many of the areas which are in red, you can see in future by nearly 2040 we face highest scarcity of water, and the crops this year we have faced tough is soybean, chocolate, where the prices have gone up. And also you can see the pictures where the water crisis is expected by 2040. This is the part in Madhya Pradesh where we are facing this challenge. And apart from that, due to this abiotic and biotic synchronization, the major diseases are going to increase, and this is such an outbreak has happened. And this is a very important slide where we can see the interaction between biotic and abiotic stress, which lead to the disease. And what are the challenges? So one of the best thing what Morning was discussing is biotechnology and genetic improvement is one of the important thing, where I will be focusing more on genetic resources, climate resilience crops, and genomics, where we can go for this. And we can see that how important is the genetic resources, genomics, and gene editing. And I will not much focus on gene editing since my subject is on genomics. I will give some importance. See, I think important to take an examples of mulberry, since I have worked for 6 years. Genomic resources play a vital role for any development of the crop. And you can see the greatest variation in this, very good diversity. And the conservation is an important task in mulberry, since it's a perennial crop. Most of the time goes for conservation, and also good thing is it can be clonally propagated. And other important thing for a breeder is use of crop oil relatives instead of bringing down the narrowing the genetic gain. And some of the techniques, crossing and hybridization is also very important, where we carry the important disease resistant genes. And you can see is the diploid, this is the decosaploid, where the chromosome number goes up to 131, 138. This is found in Turkey. Best example, the fruits are stored in the market. And as the revolution passes, currently we are in many sequences have been, many species have been sequenced. And if you see over the years, recently we lost James Watson, who is Watson, where one of the pioneer in genome. And here also we can see the currently we are in 4th generation of sequencing, which is very important. And currently it's very easy to sequence compared to the other, because due to the HiSeq and other sequencing long reads was available. And this is where different platforms are available. In mulberry, all the 3 different species are being sequenced. You can see very good idea of information on mulberry. And these are very important for genotyping. As we have developed a beautiful protocol using the meristem technique. This helps to directly give the chromosome counts. You can see here in one of the published papers, where previously it was very not clear. Currently, with our protocol, we can count the exact number of chromosomes using the probodium iodide method. And in mulberry, you can see the variation from diploid to hexaploids, and also correlation between the ploidy and pollens. And coming to the mapping, one of the important for developing is climate resilient crops is identifying the genes or traits related to the particular important. Where I was working in Canada on stem breast, we could develop a diagnostic markers where it helps to develop more resistant or tolerant varieties and see beautiful maps with 4 populations, and diagnostic markers can be used to screen the entire germplasm lines. Similarly, with this, we can screen resistant and susceptible genes. And also, one of the important factor is association mapping, where you can target the genes. And from this available information, you can go for genomic selection and future for lays for gene editing. Gene editing is one of the important thing where I'll tell a few words about that. Now the trend is pangenomics and super pangenomics, where one of the reference genome is not sufficient. We want multiple reference genomes to identify the genes of our interest. See here, we can see here beautifully with one reference, we can't identify the genes associated with the disease, but using the pangenomes, we can easily identify the very important genes related to the interest of our trade. Here they have targeted for downy mildew. And again, genome editing is one of the techniques very gaining high popularity, but currently in India we don't have this technology. This was like one of the by 2050 will revolutionize if we use in India, and more than 60 crops we have the techniques and they have developed the lines for gene editing. And future, what is the future in genomics? Developing sequencing information, market development, screening, and so on. And coming to the precision farming, where the next already is taking a shape in India with the help of the support of artificial intelligence and machine learning. We are already using in our Krishi Vignan Kendra. And what is the important thing is due to the high cost, labor intensity, and also input overuse, the fields are becoming soil degradation, and also the expenses occurred to for the cost is increasing for any crop. I'll take an example of banana. And due to the use of precision farming, reduction of fertilizers up to 25 to 50%, and also the reduction in irrigation water use, an average saving cost will be around 25%, 10 to 15% saving labor charges. So Japan leads the way. Currently in India also, we are on average 40 to 45% of water is saved. And the main component of precision farming is reduction in chemical leaching, enhance soil health, and conserve farm resources. Here is the picture where the yield has been increased with an example in banana, and savings up to the 20% we got, and 50% of storage of NPK can be reduced because overuse of fertilizers can lead to the major diseases. And this you can see that NRCB. Similarly, we have replicated in our KVK for banana, dragon fruit, and butter fruit. And this is a PIDA where we are targeting to export our Nanjangud Raspale. And banana is ODOP crop of Mysore. Also, as spoken by Dr. Manjunath, we are giving more importance for Panama wilt, which is a serious disease against in Nanjangud Raspale. Overall, again, one of the another use is drones. We are already using in our KVK on 18-acre basis trial. This is the fertilizer use efficiency in nano urea, nano DAP, etc. And it has come well. And also, under my leadership, we have trained 225 students using the drone and for 15 days training. So this is where our Honorable Minister, Central Minister visited our farmers field during this visit when he came to Mysore. And overall, we don't have this technique currently. AI, robotics, and automation. In future, it will be coming into the field. With this, another thing is use of conventional way of natural farming or organic farming to bring back this soil. So these are the, if we don't take care now, these are the crops we should eat in near future. With this, some of the books we can refer. Recently, they will be released in January 26. Sustainable Agriculture Next Generation on crop production, crop improvement, and crop protection. So thank you for this wonderful opportunity. Thank you, one and all.
Thank you, sir, for the nice presentation. So when it comes to precision agriculture, so normally data has to be fed or
No, we get every 15 minutes we get the data. You can get the data when you suppose I'll give you an example in banana. There will be a weather station with 13 parameters it will be giving relative humidity, rainfall, wind speed, everything. So the data is collected in the field. It will go back to the server, then to the company, and again back to our SIM mobile. So over the days we can calculate what is the percentage of water intake is there because all it works with the sensors. Soil moisture meter will be there, pH meter will be there. What is the pH of the soil? And if you go in advance in Baramati, where farmers are so advanced, they are taking the help of uh, remote sensing the satellite images, where they precisely give the irrigation to the exact point where the need irrigation in sugarcane. Similarly, the spraying also, where the field is deficient in nitrogen, they go for the application of the particular field. That is, 3 companies are working. One is for soil testing, and they have to just pay 10,000 for the field, and the data is taken care. And here, one thing in banana, we can control the irrigation. Wherever we sit, we can control the irrigation. And the solar energy will help us to give the data or feedback because no electricity is needed to take the collect the data. So more data we will get rather than giving the data. Just we have to tell when to irrigate, how much time we have to irrigate, we can feed it, and through the app we can monitor completely entire field.
So it means you need to have connectivity, internet.
Yes, 100%. It's completely works on IoT, Internet of Things. This we are already using in our KVK and also robots. Only thing we are lacking behind is robotics. So next we have to try that.
Okay, all the best.
Yeah, thank you very much.
Next, Shashidhar. Next speaker is Shashidhar.
Good evening, respected dignitaries and delegates, and every one of you here. 15th one. Okay, my presentation is on impact of agri-nutri gardens on food, nutrition, and livelihood security of farm families. So, coming to the introduction, so as we all know, agriculture is the primary livelihood security of all the South Asia. So, at the same time, in this region, it houses large enough population of undernourished peoples. So, there is a need to look at the multiple strategies. So, in this context, so agri garden is a sustainable framework in order to reduce the malnutrition as well as in order to supply a constant supply of fruits and vegetables. So in this context, so it helps to improve the food nutrition and livelihood security of household or the community level. So the Nutri Garden envisages developing and demonstrating the sustainable framework to improve the health nutritional outcome. So it can be scaling up the wider adoption in rural areas. So coming to the methodology, so this study was selected in the Nanjangud Taluk. So in 2 villages we have selected. One is Hattale and another one is Jimaralli. So in this study we have provided critical inputs like seed kits like from Indian Institute of Horticulture Research. In that seed kits, various seeds are available. That is spinach, fenugreek, coriander, radish, amaranthus, lady's finger. So almost all the whatever the need of the family, almost all the seeds we have provided. So along this perennial crops also provided, that is drumstick, lemon, papaya, lime, and curry leaves. So perennial plants we are asked to plant in the buds. Okay, so we have provided the different seeds in different seasons like Kharif, Rabi, and summer. So along with we have provided perennial and medicinal plants also. So this is a methodology, that is selection of the site in the farm families and layouting of the nutri garden and giving nutrition education to the farmers and women and follow-up visit, and that is harvesting of the vegetables and the continuous utilization of vegetable, fruits and vegetables by the farm women. Coming to the results, so these are the socio-demographic characteristics of the farm families. Total, we have selected 30 farm families. So around on average 180 members from 30 families. So almost all of the farm families were belongs to a young age group, followed by, and most of them were illiterate. Okay, so education level was very low, and their family income is they belongs to low family income group. Around 73% of the families were belongs to the lower income class. So coming to the nutritional status of the farm families. So as we observed, majority of the farm families are belong to the normal nutritional status. That is body mass index. That is around 74.44 percentage. So remaining are belong to the undernourished category. So coming to the adequacy of food intake before and after the Nutri Garden. So as we can observe here, so before the Nutri Garden intervention, so most of the respondents were consuming less fruits and vegetables when compared to the recommended dietary allowances. So after the implementation of Nutri Garden, so it is significantly increased. You can see the percent increase. So especially in the fruits and vegetables consumption, around 17.4% in green leafy vegetables, other vegetables around 14.9%, and fruits significantly increase in the fruits consumption. So that is 47.1%. So coming to the impact of NutriGuard on nutrient intake of the for women. So here we can also observe. So there is a by consuming the regular consumption of fruits and vegetables, so their nutrient requirement also increase after the implementation of NutriGarden. So you can see before the NutriGarden implementation, so their nutrient consumption was very less. So after the implementation of NutriGarden, so, by the regular consumption of fruits and vegetables, so their nutrient intake was significantly increased, especially fiber, calcium, and iron and vitamin C. We can see that. So there is a substantial increase in the iron and vitamin C contents, around 22.13 and 24.98 respectively. So this is made from reference, that is ICMR NIM in 2024. So this is one of the press coverage that we have implemented this program in the Nanjangud Taluk. So coming to the conclusion, so by this presentation, I would like to say that implementation of NutriGarden in the rural areas is a sustainable framework to improve the health and nutritional as well as livelihood security of farm families. So especially in the areas where the people are following monocropping system, so they are deficient in one or the other nutrients. So in that way, these NutriGardens can be helpful in providing regular fruits and vegetables. So that indirectly increases the nutrient adequacy. So especially vital nutrients like iron, vitamin C, and fiber. Thank you.
Okay, congratulations, Mr. Deepak. Thank you, sir. You have done good work. Wish you all the best. Thank you, sir. Any question? Okay. So remaining, I think we have still 8 members to be presented. So because of the time constraint, so Shashidhar is here, no? Yes. Then Srimati Divya. Srimati Divya is there. Yeah. Okay. And Sushmita CH. Is not. Deepti. Can you present it tomorrow? Tomorrow because today only one. Deepti Madai. Want to present today? Tomorrow is that okay? Because otherwise we have to reduce the time actually if you want to present today. Okay. Okay, you can start. Sir, I think we can give 2 minutes. Yeah. 3 minutes.
Yeah. Okay. Good evening, all. Animal husbandry is a part of.
So I request you to present the gist of your work.
Yes, that's what it needs a background, otherwise it cannot be understood. Okay. So animal husbandry is a part of agriculture. Presently, we know that there is a huge demand for the fodder in agriculture, whether it is for the milking cattle or for intensive rearing of animals like cow, goat for meat demand. So we need more fodder. And presently there is a lack of sufficient production of fodder, and moreover nutritional fodder is lacking. So we are using only cereals like maize, sorghum, and other things, and they are lacking nutritious fodder. So in this regard, so we need pulse fodder crops. So among them, if you take the arsenal, it's not much, and other crops. So cowpea is the major one, which is producing maximum green fodder. Okay. So now, because of this, so there is already 2-3 varieties of cowpea for fodder purpose, but we want to improve on them. So we want to release new varieties which are going to yield higher biomass and also more protein content. So in this regard, we know that we require the genetic resources. That is, we have to collect the germplasm and evaluate it. So in this regard, so we have, so this is the brief I can do on. So we want to evaluate some germplasm lines and use it as a parentage in crossing and development of the new varieties. So nearly the 72 germplasm lines along with the new advanced breeding lines we have utilized in screening to develop high yielding varieties. So we have screened in 2 seasons. One is in Rabi, another in Kharif. So these are the 72 lines, and these are the release check varieties. We want to see whether any lines are there which perform better than checks already released varieties with respect to yield as well as protein content. And when we analyzed, means when you grow crop and recorded the data on these parameters, and we come to know that there is sufficient variability among the genotypes used for different traits, so that we can utilize these lines to develop new yielding varieties. So you can see, so we have these are different parameters, and heritability of those traits are there. So this is where the estimates of mean sum of squares, and you can skip it. So this is a pooled analysis of ANOVA, means both the seasons. So Kharif and Rabi, we have pooled the data, and we have seen that there is sufficient variability among the genotypes for all the traits. And next is, so there is sufficient variability, but we want to select as a parentage. So genetically similar parents, we cannot choose it, but which one we have to choose for parentage? So we have to club it, similar types we have to club it. For that purpose, we have gone for clustering, wherein among the genotypes are clustered depending upon the similarity, so that we can select the lines which are genetically dissimilar, means they are falling apart. That is cluster 1, green fodder is low, cluster 8, genotypes which are having this higher relief, so that when you cross it, you can get better progeny, that is F1s, and higher in this. We also estimated correlation for green fodder yield. What are the traits responsible? So there is a fresh leaf weight, fresh stem weight, and the leaf ratio are major important. And when we estimated among 72, the 4 lines in the Rabi and 6 lines in Kharif, they have shown more protein content and also high green fodder yield compared to the checks. So these lines can be utilized as a parentage development of new high yielding varieties for fodder crop. Thank you.
Thank you, sir. Is it ACRP work? It's part of the ACRP?
No, this is my MSc student work conducted at ASRP in ZS.
Okay, okay. Thanks, sir. Thank you.
Thank you.
Mentioned, can you go to the spacing?
Spacing? So this is a paid row. I have gone in paid row.
And 60 to 30 centimeters.
Yeah.
Correct? The recommended one.
This is for evaluation. These are genotype evaluation, not for commercial growing. So when you go for evaluation, so you want to this thing to be seen, so you have to go for more wider spacing. No, that's fine. Oh, I have an answer. See, for normal cowpea, vegetative growth is less. So when you go for fodder copy, so vegetative growth, you need to give more space. That's what I'm telling. So that is for regular this thing comes. Okay. But because we are going for paid row, so we are calling this thing. That's the thing. Because whatever side is fine, but ASFM copy is fine. Like you, but it is for fodder purpose. This recommended this thing.
Let it be checked.
Yeah. Okay. Thank you.
Next time, madam, please. Thank you, sir. 17th. So Sushmita will present, right? Yes.
Good evening to all. I am presenting my work on red rice varieties. Actually, Mysore, the major crop of Mysore is paddy. And the major variety of Mysore, the farmers are growing is Jyoti variety, because the red rice is rich in anthocyanin, and it slows the lowers the blood sugar, and it's rich in high antioxidant, iron, magnesium. These are the good qualities of red rice. And the variety, as I told, the Jyoti, the prevailing variety, it is very older variety. It was released during 1974, but still farmers are growing this variety because it is having a good market. It is export it is having a ready market at Kerala. Most of the 80% of the farmers are growing this variety, but the yield level is decreasing day by day because of its susceptibility to diseases and pests. These are all the major diseases which can be observed in this variety. So the solution for this problem is we can grow for alternate varieties, and I took a study on these different varieties. These are the different varieties. That is KMP 220, which was released from Mandya, and Sayadri Kempumukti from Shumaga University, and Shreyas from Kerala Agriculture University, and Poundami. These are all the different varieties I took for this study. And the major objective is to study the growth and yield performance of the red rice varieties during Kharif and summer season, and the farmers, and also in research plants. And these are the observation I took. And this observation, coming to observation, in the height, the KMP it is very taller than the other varieties, and panicle length is also long, and the number of panicles per seeds also it's very high, and the number of tillers is also high. And coming to yield, KMP performed very well, and it has given 130 gram per plant in both summer and kharif season. And the crop duration and KMP is very longer than the Jyoti variety. It is nearly 130 days. It took 130 days. And coming to yield, the yield is very high in case of KMP, and Sayadri Kempumukti is on par with the KMP variety. But coming to seed market, when we come to for marketing, the commercial seed rate was nearly 2,400 per quintal in case of KMP 220, and in case of Sayadri Kempumukti, it is 2,600, which is same as that of the Jyoti variety. If you see the BC ratio, the BC ratio is very high in case of the Sayadri. In case of the Sayadri Kempumukti, that is 2.41, and the farmer accepted this Sayadri Kempumukti variety during the field day. This is the plant height. The KMP gave good fodder yield also. And this is the BC ratio. Sayadri Kempumukti shows highest BC ratio. These are the observations taken during 2 seasons. This is the observation taken in farmer's field. And these are the 5 varieties. You can see the varietal difference here. These are all the seed parameters which we have taken. This is the field day conducted at KVK, and took farmers' opinion during the field day. And this is the press coverage. And coming to conclusion, Sahyadri Kempumukti got same market value as such of Jyoti. But if we compare for yield, the KMP 220 is very good. But overall acceptance by the farmer is very important. So farmer gave first rank to Sahyadri Kempumukti and second to KMP 220 and third rank to Jyoti. And they rejected the Purnami and Shreyas since its grain shape duration is not matching with the Jyoti variety. Thank you.
Speaker is Sushmita. So Deepthi Madai is here. Okay, fine.
Good evening, the chair and one and all present here. I am Sushmita from Bangalore University, presenting under the theme Advances in Biological Science, Traits in Botany, and my research title is in vitro study on glucose utilization capacity of bark methanol extract of Syzygium hemisphericum using isolated rat hemidiaphragm and its major phytoconstituents. Yes, impairment in insulin-stimulated glucose uptake is one of the major hallmark of type 2 diabetes mellitus, and Syzygium hemisphericum is one of the less known species of Western Ghats and have documented medicinal potential in traditional system. This study evaluates its glucose utilization potential and metabolic composition. This is the methodology I followed. Here, the bark was shade dried, powdered, and extracted with methanol. Then the extract of 10 mg per ml was tested for glucose uptake using isolated rat hemidiaphragm in vitro. Insulin and quercetin used as reference standards, and glucose levels in the medium was measured using Godboard method, and this SHBM was further analyzed using GCMS and HRLCMS with the positive electron mode. Coming to the results of my study, here the Syzygium hemisphericum bark methanol extract showed significantly higher glucose uptake than the negative control and exceeded the activity of insulin. In the presence of insulin, it has enhanced the glucose uptake activity, and the GCMS analysis showed the presence of 16 major compounds where octadecinnamide was predominant, and the LCMS study, HRLCMS study showed the presence of phenolics, flavonoids, and alkaloids where terpenoids, where gingerol and camphorol were found were the predominating compounds in the bark methanol extract. Concluding my topic, the Syzygium hemisphericum bark methanol extract significantly enhanced the glucose uptake in a rat hemidiaphragm, and which indicate a strong antidiabetic activity. This comprehensive metabolite profiling, both the GCMS as well as HRLCMS profiling, revealed multiple bioactive compounds with the potential antidiabetic effects, supporting the use of Syzygium hemisphericum for managing type 2 diabetes mellitus. Thank you.
So among the constituent, which was the dominant one you mentioned? That
Camphorol.
camphorol.
GCMS octadecinnamide was the predominating compound.
Okay. All the best.
Thank you, sir.
So next speaker is Ankit Anand Sinha. Is here? Today only it's fixed. Yes. So next four like Ankit, Arul Kumar, Kyadagi Ravi Kumara, and Shamraj will present tomorrow. Okay? Hope it is best.
Good evening, delegates and all the people assembled here. My name is Dr. Deepthi, and the topic of my presentation is in silico identification and functional evaluation of antimicrobial peptides from piper nigrum black pepper as potential biocontrol agents against phytopathogens. So, antimicrobial peptides, as we all know, are proteinaceous compounds which are mainly produced by plants as a part of their defense against pathogens. So, my work mainly, the objectives of my work included selection of the specific antimicrobial peptide candidate from the black pepper peptidome using a suitable deep learning model and solid phase synthesis of the specific antimicrobial peptide and analysis of its biochemical properties to assess the efficacy of the silver and the copper doped zinc oxide nanoparticles which were conjugated with this antimicrobial peptides as a biocontrol agent against Phytophthora capsicum. So the background of my study was as follows. For the deep learning model which we used, so we collaborated with the two scientists, Dr. Subudhi and Badri, and his research scholar, Gokul Chauhan from IIT Jammu, and we developed a deep learning model. The overall workflow is as follows. The AMP prediction was done by the machine learning models from which we identified the suitable antimicrobial candidate, after which we synthesized by solid phase peptide synthesis. Then we prepared the nanobioconjugate with the AMP and the silver and the copper top zinc oxide nanoparticle, evaluation of its biocontrol efficacy, and various assays were conducted. So this was the PyPPEP database which was mainly used for my study. It is a manually curated database consisting of experimentally derived peptides from the piper nigrum, from which we developed the deep learning model. It is a BioLSTM model, and the various data sets which were used were obtained from for training the model were from LAM, PiperPep, Uniport, and an independent data set. So from this, we developed the data set was collected, and this is the model architecture which is mainly used for obtaining the ideal AMP candidate. It follows a BioLSTM model from which the model was created, and after which the performance of the model was evaluated based on these 5 categories. So it was found that our data set and using our BioLSTM model, a good accuracy and precision was mainly obtained, and the amino acid sequence highlighted here was the one which I selected for my work. So this is the experimental procedure. So this is the piper plant which shows a lesion infected by Phytophthora capsicum, from which we isolated the RNA and the formaldehyde agarose gel electro was performed. After which the PCR and cDNA extraction was done and it was sent for sequencing. After sequencing, the data, the amino acid sequences were then converted to peptide sequences, and it was used in plastics, and it was confirmed that it was a good predicted antimicrobial peptide. So this sequence was mainly used for solid phase peptide synthesis from GenScript with a purity greater than 95%. So then we conjugated the peptide with the nanoparticle, and this is the STEM and the TEM analysis which was performed of the particular nanoparticle conjugated. So then this was the FTIR analysis of the nanoparticle conjugated with and without the peptide, and the analysis proved that there was a good binding between the peptide and the nanoparticle. Similarly, UV visible spectroscopic analysis also showed an increase in the wavelength absorbance, indicating good binding. Then photoluminescence spectroscopy also revealed a good binding between the nanoparticle and the peptide. So this shows the lesion size of the particular fungus and during various treatment, and it was found that the infectivity assay showed that the copper doped nanoparticle with the peptide, no lesion was obtained in 24 hours. But 48 and 72 hours, the lesion size was seen, but very less diameter. And this is the microscopic analysis of the particular. So here, trypan staining was done, and it was observed the fungal hyphae was seen in the control leaves, whereas in the peptide treated leaves, even though the fungal hyphae was seen, the quantity was less. So currently, I'm working on doing the various other staining methods using cell analysis also. And finally, the in vitro analysis of this particular nanoparticle conjugated peptide in the media was done, and as the concentration increases, that P capsicum growth was decreased. Thank you.
Okay, all the best. Presentation was nice. So by this, I think we will conclude this session because the already it is now 6. We have crossed 6. So as per the direction of the organizing chairperson,
So we have to complete this and remaining people you can morning session. Is it okay, sir? Okay.